Bhujwar Knowledge City, the part of MIT League of Colleges, driven by a single-minded focus on imparting quality education. MIT BKC Institute of Pharmacy established in 2006 with a mission to develop students socio-technically. The institute has maintained the quality right from the establishment, as it reflects from the accreditation and various awards which college has achieved. MIT IOP is the first NBA accredited pharmacy institute in Nasik region. MIT BKC Institute of Pharmacy has been approved by AICTE PCI and is affiliated to Savitribai Phule, Pune University. MIT BKC IOP offers full-time courses in Bachelor of Pharmacy, Masters of Pharmacy and PhD. Institute stands for its excellent, well-lighted and ventilated ambience with range of facilities like classroom, which are equipped with an overhead projector, speakers, smart board, etc. Well-equipped and spacious laboratories which provide experimental learning experience. State-of-art pilot plant simulating the pharmaceutical industry with range of equipments for formulation, development and evaluation. Institute has separate instrument room having range of sophisticated instruments as per the need of industry and research work like HPLC, UV, FTIR, etc. Heart of Institute, the library, one of its own kind with open access, great ambience and various national and international book collections to give great learning experience. Institute is also a well-with gymnasium which is equipped with a number of professional electronic and manual universal fitness machines along with indoor sport facilities and music room. Campus also has an amphitheater for social gathering and expression of students. The institute believes that the quality of faculty member and their commitment to excellence is the core competence towards its development. With this thought, institute have more than 50% doctorates, 200 plus publications, 5 patents, 1 copyright and 16 book publications. Our students are encouraged to take part in national and international competitions and events as part of their curriculum. To add feather on hat, we have Miss Archana Kapse, Air 3 in GPAT 2019, Mr. Aditya Darade, Air 1 in GPAT 2014 and Mr. Parag Mehta, Air 15 in GPAT 2010, who have qualified national level exam GPAT with dignified ranks. MIT IOP has signed 8 Memorandum of Understanding, that is MOU, amongst them are Japanese Industry, Asahi Kasei and Malaysian University Technology Mara. MIT Institute of Pharmacy provide a platform for expressing students and showcase their hidden talent through Antaranga and MIT Utsav annual social gathering. Success comes down to you with appropriate attitude, effort and focus, which is certified by various achievements which Institute has received. Best Magazine Award by SPPU, Champions Trophy at National Pharmacy Week, Award for the Best Research Paper and Presentation, Best College Award by Student Development Board SPPU for Student Welfare Activities. Institute has also been honored with highest rank platinum by AICTE C2 survey for its outstanding linkage with industries for consequently 2 years, which surely strengthen the institute and students to survive in this brave new world of technology. Bhujbal Knowledge City Institute of Pharmacy, the place where your search ends.
So, good morning. Myself, Ms. Anjali Tajanpure. Welcome you all for the one-day national level webinar on Introduction to the Current Trends in Experimental Pharmacology, organized by IQAC Cell and Department of Pharmacology, Mumbai Education Trust, Institute of Pharmacy, Bhujbal Knowledge City, Adgaon, Nashik. It gives me immense pleasure to welcome you all to our institute, which is marking 30 years of excellence with the generous support of and vision of our Honorable Chhagan Bhujbal sir, and also with the continuous support of Trustee Honorable Sameer Bhujbal sir and Pankaj Bhujbal sir. Also, we get the continuous support from respected Shefali Bhujbal ma'am, which is Chief Administrator of MET BKC. The institute started the journey towards the academic excellence in 2006, and it is approved by AICTE and PCI. Our institute is affiliated to Savitribai Phule Pune University. Our institute is accredited with NBA and NAC2. We have been honored by platinum rank by C2 survey for industrial and institute interaction by AICTE. The institute stands 76th in the band of Pharmacy Institute of India by NIRF ranking in 2020. MET Institute of Pharmacy provides various courses like D-Pharm, B-Pharm, PharmD, M-Pharm in subjects like pharmaceutics, pharmaceutical chemistry, quality assurance techniques, and pharmacology. MET IOP also provide course of PhD in pharmaceutical sciences. With this corporative beginning, MET is redefining this system of education to meet the current need of market. The webinar is a form of institutional support that institute and academic department provide to the students and faculty to get them updated to the current need. Thus, with the above objective, METians have made an attempt to explore the new trends in experimental pharmacology to develop professionally and technically sound students as well as faculty in the pandemic situation of COVID-19. So, to move the session ahead, I would request our conveners of today's webinar, Dr. Pawan Udavan sir, to take the charge over. Over to you, sir.
Thank you, madam. Good morning, all respected dignitaries who are present on this virtual platform. Respected Principal Dr. Sanjay Ksirsagar sir, Chief Administrator of the campus, Honorable Dr. Shefali Bhujbal madam, very honorable and respected Chief Guest of the function, Dr. Subedar sir, and all the respected dignitaries. From the outset, let me welcome you all on behalf of my principal, Dr. Sanjay Ksirsagar sir, and Mumbai Education Trust's Institute of Pharmacy, Bhujbal Knowledge City, Nashik. Very nice to see gathering on this virtual platform. And as we all know, this is a new normal, but the knowledge should be on a continuous mode, as a part of which the IQAC and the Pharmacology Department have organized this national level one-day seminar on introduction to the current trends in experimental pharmacology. We all know the importance of drug discovery and searching for the better and safer drugs, particularly in this pandemic situation. And we, the Academic Institute of Pharmacy, we thought that we should also come ahead and motivate the colleague pharmacology professionals. And for that, it is very essential to get aligned with the current trends and the techniques and tools that are used in experimental pharmacology. As a result of which, today we have organized this webinar, and let me tell you that we have very resourceful and honorable persons with us today. To start with, we have a chief guest, Honorable Dr. Subedar sir, who is working in ICR, and we have lectures to be given by Professor Dr. Sadhana Sate, who is from ICT Mumbai. We have Dr. Aniket Mali, who is working in National Center for Cell Science, who is going to talk on the cell cultures. and applications in drug discovery. We are also going to be joined by Dr. Kalidas Kohle, who is working in TIFR and who is going to talk on zebrafish and its application in pharmacological and toxicological screening. So from the outset, I once again welcome you all, and let me now take an opportunity, and because it is an extremely honorable moment for me to introduce the chief guest of the function. So let me quickly introduce Honorable Dr. Subedar sir, who is the chief guest of the function. So, dear friends, respected faculty members and students, Professor N.K. Subedar, who is working in Indian Institute of Science, Education and Research, was previously working with Nagpur University Department of Pharmacology and retired in 2008. Sir has various grants to his name and received various funding from DST, DBT and various reputed agencies. Sir has bagged many awards and honors. To name few, Sir has the Auslon Prize by the Journal of Peptides. Sir has won the Busseri Award from the Royal Society of London. Sir is a fellow. Sir has won the Fellowship of Maharashtra Academy of Sciences. Sir has also received a fellowship from National Academy of Sciences from the central government. Sir was former president of the Indian Society of Comparative Endocrinology. This is just a few lines, but Sir has bagged many awards and honors to his credit. Sir has guided 15 students, and 15 student has been awarded PhD under the guidance of respected Subedar sir. Sir and his team have been studying the role of some neuropeptide transmitter on the processing hunger, anxiety, depression, learning and memory, reward and drug addiction related information. Sir is ad hoc reviewer of several international journals. Sir has delivered invited talks in several institutions and universities, not only in India but abroad as well. Sir has participated in several scientific meetings in India and abroad. Sir is a reviewer of research projects which are submitted to the funding agencies like SERB, CSIR, UGC, DBT, and many. And out of India, sir has been reviewing the projects that are submitted to Portuguese Foundation and the Schrodinger Fellowship of Austria. Sir has published more than 130 research paper in reputed international journals. So this is just a brief introduction to the respected Honorable Subedar sir. Now I request Subedar sir to virtually inaugurate the function. Sir.
Uh, thank you very much, Professor Udavan, for that wonderful introduction. There was a little bit of exaggeration. That's okay. A bit of exaggeration. Okay, good. So let's begin. Okay, Honorable Chef Ali Bhujbal Madam, Dr. Shirsakar, Dr. Udavant, Sadhna Sathe Madam, Mali Sir, and the faculty at the Institute of Pharmacy, students, friends, and colleagues. Um, First of all, I'm very impressed to see the progress made by this institute over the past about 40, 50 years or so. It has really taken big strides, and I'm very optimistic that it will make a commendable progress in the time to come. And I look forward to that. I'm very happy to be with you this morning and be a part of this exciting deliberations on the national level one-day seminar on the introduction to current trends in experimental pharmacology. I feel that this is a perfect moment to undertake this exercise. Experimental pharmacology is extremely important for two reasons, in my opinion. First, it not only tells us about the action of the drugs, but also about the biology on which the drugs are interacting. Indeed, pharmacological agents have greatly helped to understand the working of the brain, which is the primary focus of my interest. Second, is a matter of immediate concern. For experimental pharmacology, we need to use rodents, rats and mice. Unfortunately, it has become increasingly difficult to work on these animals in view of the unreasonable restrictions imposed by the authorities like UGC and CPC-SCA. I believe that we all experimental pharmacologists need to raise our voice in support of the use of these animals in our search for newer drugs and towards the service to humanity. As the science keeps on progressing on different frontiers, different things are happening which are extremely relevant to experimental pharmacology. I would like to take the opportunity to talk to you about some of the cutting edge news technologies which are extremely relevant, and particularly the younger pharmacologists should pay much attention and focus towards these. One such method is optogenetics. Now, before I talk more, some of you already heard about it, but for the younger faculty and students, I would like to comment. I would like to show you a small video clip. It's only a very short clip, which I have got from YouTube, but let us see the clip and understand the science behind it. I think this is an amazing way to do science. Can I request somebody to please switch the clip, please? Please take a look at the mouse. We are looking at the behavior of the mouse, and you will find that the behavior of the mouse, now it is not eating. Now you see there is a see very carefully. There is an electrode implanted on the head skull of the mouse, and you can see the light there. The light is exciting certain part of the brain, and it is eating. It is eating. The light is on. Light is on. Just keep on looking at the light. Now the light is off. Once the light is off, the animal has lost interest in the food. It is not interested in the food. Not interested in the food. Just keep on. Now the light is on. Once the light is on, you will find that the animal's interest in eating has again started. So the animal keeps on focusing, keeps on eating. So as if by exciting certain regions of the brain, you are able to control the behavior of the animal. The animal is eating because the lights are on. When the lights are on, the certain areas of the brain are being excited. When they are being excited, now the lights are off. Look at the animal. The lights are off. Once the lights are off, the animal loses interest in the food. Don't you think this is an amazing technology? Let us briefly see what is really happening. The science behind this is very exciting. There is an alga called as Chlamydomonas, and since it's an alga, it does photosynthesis. And to facilitate photosynthesis in its plasma membrane, it has certain channels, and those channels are sensitive to light. So what the scientists have done is that they have removed the gene which is responsible for that channel protein, and they have introduced that gene into the DNA or into the chromosome of the mouse. So this is a transgenic animal which has received a gene from an algae. Now this transgenic animal, now that gene is everywhere in all cells of the brain. Now what then they have done is they have implanted a laser fiber that specifically eliminates that part of the hypothalamus where the cells or where the neurons are, which encourage the animal to feed. Now, as long as the light is not on, those cells are inactive and the animal doesn't feed. The moment we switch on the light, those specific neurons in the hypothalamus are excited and the animal likes to feed. Now, as long as the light is on, those neurons are excited and the animal keeps on feeding. The moment you switch off the light, the neurons are no more excited and the animal stops feeding. This is an amazing technology. This technology has been used not only for the mouse, but even you have zebrafish and Drosophila, which have been which can in which the neurons or different parts of the body can be excited by means of laser, and these are amazing techniques to tell us more, not only about the activity of the cell, but how the drugs can motivate this activity. So I thought this is a very exciting way to start to acquaint the younger faculty about this, and many more such techniques are arriving on the line. And now it is the challenge of the next generation pharmacologists, particularly the experimental pharmacologists, to adopt these techniques, to learn about them, and to use them in your own research. Then only, let me emphasize, then only India will be able to compete with the research that is happening elsewhere in the body, elsewhere in the world. So finally, I would like to say that this has been great opportunity and experience for me to visit your institute, maybe virtually. I look forward to an opportunity to visiting your institute in person as COVID permits. And I once again wish great success for this amazing workshop, and I'm sure you will all enjoy and benefit from this exercise. Thank you so much for your interest.
Thank you so much, sir. Now I request Honorable Subedar sir to virtually inaugurate this one-day webinar. Sir.
Okay, thank you very much, Dr. Udavan, for giving me this opportunity to inaugurate. And I hereby inaugurate the conference on experimental pharmacology. Thank you so much.
10, 9, 8, 7, 6, 5, 4, 3, 2, 1.
So now Chief Administrator of MET BKC Nasik, Honorable Shefali Bhujbal Madam is going to present a keynote address. I request Shefali Madam to kindly address all of us. Over to you, madam.
Good morning, Subedar sir. Good morning, Shirsagar sir, and my colleagues and my team of pharmacy, Met Institute of Pharmacy. This time, when we are inaugurating this function, little bit of pharmacology I have come to know due to Subedar sir, and how the clinical trials are being taking place in the industry as well as in the institutes. So from last past year, due to this COVID-19 pandemic, people are aware about certain words like clinical trials, drug discovery, and further few more words about it. So now, as we are aware about all these systems and all these clinical trials and all these discoveries which has been done in the whole world. So now, as we come forward with this maybe a new research, because there are various variants of viruses, maybe bacteria, or maybe some few more diseases which we have never heard, and we are hearing for the last whole year. So for this, as an institute as well as an industry of pharmacy, we have to upgrade ourselves into all these new developments of pharmacology, and we should be focused on developing more and more processes which has a typical platform, a uniform platform and discipline in the preclinical stages. So as an institute of MET, we are always very much sure and very much upgrading ourselves in each and every verticals, because through maybe some support of the industry and as well as with these kind of workshops. So I am sure, and I urge the industry, the pharma industry, as well as the institutes like our own institutes and other institutes, to think about this subject and think about all these drug development and clinical trials as a very very focused subject, which can be an example, set an example in the recent drug developing formulas, and as well as more and more institutes and more industries can come forth and forward to think on these lines. And I'm very much sure with this platform, as we are conducting this seminar, the whole day seminar, and I'm very much sure that the pan India people who are connected to us might get few more steps ahead how to go ahead with this clinical studies. And I'm very much sure that the whole team of MET Bhujbal Knowledge City, Shirsagar sir and Subedar sir, we can come together and we can think about more and more systems developed in this clinical trials. Just to think about and just to put a picture about how Nasik is growing, in this year we are thinking about a cluster of pharma somewhere near 30 kilometers away from Nasik, and there are various industries and institutes which are coming forth for developing these pharma research orientation systems. And I am sure that you people can take part into this, and we can think about more and more clinical trials to perform and have patents to it, and actually it can be used on humans to treat these diseases. Thank you very much for calling me on the inauguration platform. Thanks a lot.
Thank you, madam. Thank you, madam.
Yes. So let me take this opportunity to present a vote of thanks for this inaugural function. So on behalf of respected Dr. Sanjay Kshirsagar sir, Mumbai Education Trust, Institute of Pharmacy, Bhujbal Knowledge City, honorable trustees, various departments of Institute of Pharmacy and Bhujbal Knowledge City, I take this opportunity to thank Honorable Subedar sir and all the delegates who are present for the inaugural function of this national level one-day webinar on introduction to current trends in experimental pharmacology. I once again thank you all, and I announce that the inaugural function is over. Thank you, Subedar sir.
Okay. Thank you, Udavan sir. All the best. Thank you.
Yes.
Thank you, Subedar sir, Shefali madam.
Okay. Now I hand over to my colleague, Professor Rahul Sable, who is going to introduce the first speaker of the day, Dr. Aniket Madi sir. Over to you, Sable sir.
Thank you, Udan sir. So, good morning, one and all present over here. Myself, Rahul Sable, working as an assistant professor in Department of Pharmacology. So, I welcome you all for this webinar. So, it is great honor for me to introduce and welcome our first speaker in this webinar, Dr. Aniket Madi, PhD in Pharmaceutical Biotechnology. Sir had 12 years of experience specializing in pharmaceutical biotechnology. Sir was worked as a senior research fellow in Interactive Research School for Health Affairs, IRSHA, BVDU, Pune, Maharashtra. In the same institute, Sir was promoted as a junior scientist in 2015. Now, Sir is working as a research associate at National Center for Cell Sciences, NCCH, Pune. Sir had number of national and international publication as well as book chapter in reputed journal like Springer and Science Diary. Sir also peer reviewer of two journals like Drug Metabolism and Personalized Therapy, Germany. Also, Sir had reviewer on Current Pharmacology Report, Switzerland. Sir was honored by many awards like Maharashtra State Board and Social Welfare Department of Maharashtra State for securing first rank among physical challenge student in Kolhapur Board in HSC exam 2002. Also, sir received Dhirubhai Ambani undergraduate scholarship in 2002. Also, sir joined NAIP ICAR 3 Senior Research Fellowship in 2012. Sir has research interest in a different topic such as translation research, cancer biology and medicine, cancer metastasis, anti-cancer drug discovery, and pharmaceutical biotechnology. So this is just brief introduction of today's speaker, Dr. Aniket Mali. So I request sir, please take your hold and start your session. Thank you. Over to you, sir.
Thank you,
Sir,
sir,
your
for very nice. Yes, yes.
yes.
Uh,
Thank you. Thank you.
can you let me know, am I audible?
Yes, sir. You are audible, sir.
Yes. Thank you, sir, for your nice introduction. Good morning, all. First of all, I would like to thank respected uh, Professor Dr. Shisagar, sir, Principal of MIT College, for inviting me as a speaker on this occasion of seminar. And I also thank Professor Udavan, sir, for and his organizing committee for organizing this event. So I am just sharing my screen. Is it visible to all?
No, sir. Yes, sir. Yes, sir. It is visible now.
Okay, so let me start the seminar. Good morning, all. So this is a topic related to the most of the interest in pharmaceutical research. So the title is Cell Cultures in Pharmaceutical Biotechnology, Applications in Drug Discovery and Experimental Pharmacology. So today, before going to direct applications of cell culture technology, I would like to just briefly introduce the pharmaceutical biotechnology field. This is a very relatively new and growing field where the principles of biotechnology are applied to the development of drugs and therapeutics. Majority of therapeutic drugs in the current market are biologics or biopharmaceuticals, which are based on antibodies, nucleic acid products, therapeutic proteins and peptides, and new generation vaccines, etc. Such biopharmaceuticals are developed through several stages similar to that of the traditional pharmaceuticals, which involve the understanding of disease mechanisms underlying the principles of health. Similarly, we have to find out the molecular mechanisms involved in the disease progression and health involved health roles of the biomolecules. Similarly, we have to synthesize and purify the molecules. Then we have to determine the processes for improving the shelf life of these products. Then we have to investigate the stability, toxicity, and immunogenicity of these biologics. And similarly, we have to identify the drug delivery system appropriate for these biologics. And last step is nothing but the patenting and clinical trials. So this is again the same procedures we follow for the whole pharmaceuticals, but the biologicals or biopharmaceuticals are also developed by the same process. But the pharmaceutical biotechnology plays important role in this development of biopharmaceuticals. So let us start with the introduction of animal cell culture. So cell culture is one of the cutting edge technology given by the pharmaceutical biotechnology, which helps in drug discovery process now and experimental biology experiments designing. So if we define the cell cultures, it's easy to define. It is nothing but just the removal of cells from the animal or plant tissues. Then we have to culture these cells in the laboratory in a favorable artificial environment, and we can carry out some research activities or experiments on these cell cultures in the laboratory. So how these activities are carried out based on the different enzymatic and mechanical processes, where it may starts from the taking the tissue from plant or animal. Then you have to segregate the tissue by deaggregation process, for which we can use the some enzymatic processes or mechanical means, and then the segregated or separated cells are cultured in the cell culture flask. These are the specially designed flasks for this technology, and these are surface treated cell culture flasks where you can grow the cells in this flask with a with by providing the nutrients and growth medium. Now these cell cultures can be converted, or these primary cells can be converted into primary cell culture. Then they can establish into the secondary or tertiary cell lines. So not necessary that every time you have to take the tissue samples for cell culturing. There are lots of established cell lines which can be directly used in your experiments, and you can derive the number of cells whatever you want from that cell lines as per your specific purpose. So, what are the basics of cell culture? So, let us start with the basics of cell cultures. It may start from the primary culture. Primary culture refers to the stage of the culture after the cells are isolated from the tissue. So, once you isolate the cells, original cells from the dissected tissue, and that cells, if you culture in the laboratory with the help of some growth medium and cell culture facility, that cell culture is referred as a primary culture. And here you have to provide the nutrient medium from outside, and these cell cultures can be subsequently transferred into the new flask. That procedure is called as subculturing or passaging. So with the help of subculturing and passaging, these primary cells can be cultured further for your experimentation. Again, this process starts from the tissue acquisition. Whatever the tissue you are interested, you can take them from animal source and or plant source. Here we are focusing on the animal cell culture. That's why I will give the examples of animal tissue culture only. So for example, we can take the cancer biopsies, and from that cancer biopsies we can dissect out the cells, and after dissections we have to remove the fatty and necrotic cells. After that, all the cells should be disaggregated with the help of mechanical and enzymatic disaggregation processes. Now these separated cells are incubated in a specific growth medium, and they are dispersed in a specific vessels. And after 24 hours the medium will be changed, and again you have to make sure that you have to stabilize these primary cells with the help of different processes, and purification of primary cells can be achieved with the help of different selective medias or different level of attachments or immunomagnetic beads. So this is the general flow of primary culture, where you can directly derive the cells from a human tissue also, and you can culture them as a primary culture cells. Second major aspect of cell culture technique is nothing but the cell lines. As I told you previously, that it is not necessary every time to take the tissues from the donors. You can use the established cell lines. There are lots of established cell lines available in the research field as well as for commercial use. These cell lines are derived from the primary cells only. So by definition, after the first subculture, the primary culture becomes a cell line. So when you establish the primary cell culture, you can convert them into the cell line. Cell lines can be of majorly three types: finite cell line, continuous cell line, and transformed cell line. Finite cell line is the cell line which is having a limited lifespan. Limited lifespan, that means every normal cell have the capacity to divide only a limited number of times before losing their ability to proliferate. After some period of time, or after some dividing, the every cell has the ability to lose its can lose the ability to proliferate, and this is genetically determined event which is called as senescence. So such kind of cell lines which are having a limited number of proliferation abilities can be called as finite cell lines. These finite cell lines sometimes can become immortal with the help of due to some processes like transformation. These transformations happens maybe spontaneously, or sometimes they may be chemically induced or virally induced, and these transformed cells can become immortal, and these cell lines which can acquire the ability to divide indefinitely, and that's why these finite cells can become continuous cell line after the acquisition of uh, properties to become immortal. And some transformed cells can also become a tumorous cells, which can be used for as a cancer cell line for cancer research. So after having a brief introduction of the basics of cell culture, then if we jump to the cell culture applications, definitely cell culture is a cutting edge technology which have a very wide variety of application, wide range of application in the interdisciplinary areas of biomedical field. So it starts from the biology, where it acts as a model system to understand the cell biology. You can understand the cells, each and every type of cells, at molecular level, at cellular level. Then it helps you to in the field of virology, then new generation vaccine productions, disease biology, genetically engineered proteins and genetic engineering field. It can also help you to carry out the research in the cancer research, then drug discovery field, experimental pharmacology field, tissue replacement, that is regenerative medicines, biomolecular discoveries, genetic engineering, gene therapy, stem cell research, genomics, and toxicity testing, and many more. They are having uses in different fields also. So if we try to focus on today's topic, then definitely we will discuss the two major areas where the cell culture have the great applications, and that are nothing but the drug discovery and experimental pharmacology. So before jumping to the direct applications of cell culture techniques in uh, drug discovery, I would like to just briefly introduce the drug discovery process. So drug discovery process is a very complicated and time-consuming process, and most of the pharmaceutical industries invest lots of money in that, and the failure rate of drug discovery is very high, and that's why the drugs become costly, and the efforts taken before one particular drug reaching to the market are tremendous, and this pipeline is very huge. It may include a number of stages of research field, starting from the disease biology. The first stage of drug discovery pipeline is nothing but to understand the disease biology, and nowadays it is important to understand the each and every disease at a molecular and cellular level, so that we can identify the targets. Second step is nothing but the target identification. Each and every disease can be easily cured if you find out the specific target or a major protein or major target involved in that disease. And if you identify the correct target, then definitely the success rate of the drug discovery will be high. But majority of drug discovery fail in this stage, target identification, because of poor target identification. Next stage is nothing but the assay developments. After identifying the target, you have to you have to design some assays, so that the molecules under research can be studied on that target. And for that purpose, you have to design some assay developments. These assays are maybe of biochemical assays. Traditionally, the biochemical assays were used, but recently the assay development stage includes some majority of cell culture based assays. In next slides, I will explain that assays. After developing the assays, then you have to go for screening of the molecules. Now, understand the process of drug discovery. It will never start from one or two molecules. It will include the screening of thousands to millions of molecules, and you have to screen the thousands to millions of molecules on the specific target you identify. And you have to identify the molecules which may be suitable or which may give the desired effect on your target. And that's why the screening step becomes a huge step, time consuming step, where the lots of money is also included, and it is not possible to carry out this thousands to millions of molecule testing by manually, and that's why the automated instruments are used, like high throughput screening. Then next stage is nothing but to identify the hits. Hits are nothing but the molecules which are producing desired effects on your target. I am saying desired effects. If you want to inhibit that particular target, then the molecule should inhibit that particular target. If you want to stimulate that particular target, the molecule under study should stimulate that target. So whatever the desired effect you are looking for, if out of the 1 lakh molecules, the only 10 to, so for example, 200 molecules are showing desired effect, that may become your hits. So again, after optimization of hits and finding out the best possible hits, then you have to convert that hits into the leads by some changing in the structures. Then these lead optimization is optimization. You go to the optimized leads have been tested for pharmacokinetic studies, pharmacodynamic studies, evaluations, and definitely once they pass the preclinical experimentation, they can enter into the clinical development, and then after passing the clinical phase 1 to phase 4 trials, then it will reach to the market. So it's a very huge process. So if we talk about the individual steps of drug discovery, the most important step is nothing but the first step, that is disease biology study, and the second step is the target identification. If we talk about the target identification, we may found its reference to the a German scientist known as Paul Ehrlich, who coined the term magic bullet. He defined that any chemical substance which is having the capability to seek out the disease or kill the disease causing agent can become a magic bullet. So, according to him, any particular drug or any chemical can become a magic bullet if we have to identify the, or we if we identify a correct target for that particular drug. So practically every medicine is interacting with the protein targets, either boosting or inhibiting the proteins, right? So for not only the receptors are also proteins, but there are so many proteins which are not receptors, but they also play important role in the disease biology. So, and that's why there is lots of science beyond receptors and lots of opportunities beyond receptors where you can make the drugs by identifying these kinds of proteins as your target for drug. So it is estimated that there are about 2 million proteins in the human body. So you can understand how much scope is there to identify the correct target involved in the particular disease biology. So yes, according to Paul Ehrlich, any drug can act as a magic bullet. In one shot, you can kill the disease only when if you identify the target correctly. So who is Paul Ehrlich? Who was Paul Ehrlich? Paul Ehrlich was a Nobel Prize winning scientist who was awarded the Nobel Prize with element making of in 1908 for his pioneering work in the hematology, immunology, chemotherapy, and for his effective treatment as a salvation for syphilis. So, according to Nobel laureate Paul Ehrlich, any drug can become a magic bullet and can kill the disease if you identify the correct target. And that's why the target identification is major important step in the drug discovery. Now, what are the different approaches we can use for target identification? Definitely, we have chemoproteomics, then transgenic organisms, then gene association studies, phenotypic screening, biomarkers, then definitely imaging techniques. These techniques we can use for target identification. After identification of target, we have to validate that target, and for this purpose, experimental pharmacology and clinical pharmacology help us to design a well uh, uh, designed experiments and to find out the mechanism of actions of that molecules or mechanism of actions of the targets involved in the disease biology. Similarly, clinical pharmacology will help us to understand what exactly happened, what is the exact role of these molecules inside the human body. So definitely experimental pharmacology will help us to identify these targets. So after identification of targets, then you have to develop the assays. As I told you, the purpose of assay development is nothing but to just investigate the molecules under study to find out their efficacy, selectivity, and potency on the selected target. And yes, I told you that drug discovery starts from thousands to millions of compounds under investigation. After initial drug discovery, the preclinical stage, only out of 10,000 compounds, 250 compounds may reach to the preclinical development. After 250 compounds, only 5 compounds may reach to the clinical development. And after successful passing the clinical trials, only 1 compound can reach to the market. So this is a huge burden on the drug discovery process, and you have to test all these molecules to find out the best suitable molecule against that target. And that's why drug discovery or assay development is most important, and it is done by the automated instruments like high throughput screening, where thousands or millions of samples can be easily tested for biological activity on on organism levels, cellular levels, molecular levels, and signaling pathway levels. So traditionally, some biochemical assays were used for this purpose, for example, which involves the enzymes, receptors, and hormones. But nowadays, cell-based assays are the major part of the high-throughput screening development, where you can use the transcriptional readouts, second messenger level assays, protein interaction assays, cell viability, and cell proliferation assays. That means cell culture technology is playing important role in the assay development step of the drug discovery. After assay development, you have to screen the drugs with the help of high throughput screening and identify the hits. I told you that hits are nothing but the compounds who are having the desired effects on your targets. So if you want to find out the hits, they may be less than 100 or they may be less than 50, but you have to identify the hits, and that hits may be a natural compounds. And after identifying the hits, you have to convert them into the leads. Now, hit to lead conversion is important because hit may be the natural compounds, and after structure activity relationships or structure functional relationship studies, you can modify their structures according to your convenience so that their safety, efficacy, and potency can be increased, and resynthesis of these molecules, small molecules in the laboratory can become possible. And that's why hit to lead optimizing is also important. And in this stage also, various cell-based assays are used, particularly cell-based assays to study the pharmacokinetics of leads and pharmacodynamics of leads are used in this stage. So if we summarize the drug discovery process and first 5 stages, that disease biology, target identification, then assay development, screening of the drugs, and hit identification and hit to lead conversion. All these stages of the drug discovery are very important, and all these stages of drug discovery are dependent on the uh, principles of experimental pharmacology. So experimental pharmacology is nothing but just test the pharmacologically unknown substances or pharmaceutical products on humans or animals. So it deals with the effects of various test substances studied on different animal species, which are aimed to the major aim of the experimental pharmacology experiments is nothing but to just identify the safety of therapeutic agent, efficacy of therapeutic agents, and sometimes mechanism of action of these drugs or molecules under investigation. So similar principles have been used in the different stages of drug discovery, and that's why the experimental pharmacology will help you to understand the principles of these drug discovery, and you can design the highly specific experiments for them. Yes, definitely, experimental pharmacology not only useful for identifying the drugs or drug discovery process, but it can be useful to identify the pharmacological effects of established drugs or already discovered drugs. So, by these two ways, you can use the experimental pharmacology for your purposes. But definitely, nowadays, experimental pharmacology is not only limited to the animal studies or human studies, because due to the 3R principles. Now, what is the 3R principles? The reason behind the 3R principle is nothing but the welfare of animals used in research is very important. So, and that's why the more than 50 years before the guiding principle is given as a 3R principle for the animal studies, which are carried out under the research head. So you have to find, you have to follow these 3R principle, and in a worldwide, scientists and pharmaceutical industries are following this 3R principle, because there are lots of ethical, legal, economical, and scientific issues when we use the animals for research purpose. So what is this 3R principle? 3R principle is nothing but the first R stands for replace. Whenever possibility is there, you have to replace your animals from the experimentation, and you have to find out the alternative methods for these assays. Second R stands for reduce. If it is not possible to replace the animals from the experimentation, at least you have to reduce the use of animals, and you have to use alternative methods for that. And third is that what the refine your experiments. Refine your experiments if it is not possible to replace the animals, if it is not possible to reduce the animals in some cases, then at least you have to refine your experiment in such a way that animals should feel minimum pain. So based on this guiding principle for animal experiments in the research, nowadays we are using lots of cell-based assays or cell culture technologies in not only in drug discovery pipeline, but also in experimental pharmacology principles. And that's why the pipeline has been changed compared to the traditional pipeline. If you here compare the traditional pipeline of drug discovery, where you can find that lead optimization and animal studies are carried out for a longer period of time, and most of the money and most of the energy were invested in animal studies in the traditional drug discovery pipeline. But due to the advancement in the cell culture technologies, where some complex 3D tissue models are used and minimize the animal studies, and you use the different three-dimensional cell culture studies to investigate the different parameters, and that's why you can enter the phase of clinical trials early compared to the traditional drug discovery pipeline. So what is the benefit of these cell culture technologies? Definitely, they reduce the time for drug discovery, they shorten the time for any drug reaching into the market, and they also reduce the price reduce the cost by 50 to 70%. So that is the biggest advantage, and that's why many scientists said that evolution in the cell culture technology actually is revolutionizing the drug discovery process. And that's why by setting up such kind of facilities at university level or academic levels, cellular lab, cell culture lab or molecular biology lab, academic institutes and colleges can contribute in the basic research, that is early drug discovery part, where you can identify the disease biology and target identification. And then you can collaborate with the multidisciplinary centers where you can test your molecules for screening, lead optimization, predictive. So drug discovery process can become easy with the help of these advanced cell culture technology use. So now let's directly jump to the actual applications of cell cultures in drug discovery and experimental pharmacology. So definitely, I already told that cell culture has been greatly evolved from two-dimensional cell culture to 2.5 dimensional three culture, that is culture in floating membranes, sandwich culture, then culture in hydrogels. Then from 2.5 dimensional to three-dimensional cell cultures, that is spheroid cultures, culture in 3D porous scaffolds, culture in 3D fiber scaffolds, then 3D bioprinted models. And nowadays, 3D bioprinting is the most sophisticated technique used to make tissue and organ constructs. So let us start with the two-dimensional cell cultures. It is also called as a monolayer cell culture. As the name defines, it's very simple type of cell culture where you have to grow the cell cultures or grow the cells on a single layer or in a single layer on a flask or petri dish containing the culture medium. So these cells are generally cultured on a single layer, and that's why the word use monolayer cell culture. They are anchorage dependent because the cells require the substrate for attachment. The cell culture flasks or petri plates used for the cell culturing are specially treated flasks where the surface is treated and that surface is used for the attachment of the cells, and that's why they are called as anchorage dependent cell cultures. These cell culture techniques are appropriate for most of the types of cells and for primary cell cultures also. They require tissue culture treated vessels, that is specific cell culture apparatus are used. Definitely these kinds of cells can be visually inspected with the help of inverted microscope. So this is an image of inverted microscope cell lines. So you can find out that easily under the inverted microscope, you can identify the cell lines as a monolayer. And then the growth is limited by the surface area. Definitely, it is based on the attachment, and that's why the growth is limited by the surface area. You have to passage them periodically. You have to carry out, you have to transfer the cells from one class to another class. That is called as passaging. And for that purpose, you can disaggregate the cells by or dissociate the cells by enzymatic or mechanical means. And that's why these techniques are easy to handle. You can culture them for longer period of time, and that you can use for cytology purposes, then harvesting some cell-based products, and obviously for research applications. So if you have an insight into the detailed scientific aspects of the two-dimensional cell culture. So if we discuss the advantages, yes, they are easy to generate and maintain. In college laboratories also, it is possible to maintain the cell cultures if you have the cell culture laboratory, and that's why they are easy to generate and maintain. They have there are variety of cell lines, and you can modify your experiments according to your purposes, and you can select the proper cell line. You can change the experimental designs and parameters as per your convenience, because they have the higher experimental versatility. Then high reproducibility is possible, and obviously low cost is there. Due to the monolayer, another advantage is that uniform exposure of drug molecule is possible. So whatever the drug treatment is given, that is uniformly applied to the each and every cell because of the monolayer. Whatever the nutrients and oxygens you are going to provide, that also uniformly applied to the each and individual cells due to the monolayer. Again, these two dimensional cell cultures can be modified slightly to become a 2.5 dimensional cell cultures by applying some extracellular matrix components like laminin, where you can convert them into the suitable interactions with the cells and in extracellular matrix components. You can also modify these assays with the help of some boiling chambers, including basement membranes, where you can study the cell migration and cell invasion activities, which are actually happening in the three-dimensional structures. But you can study these activities in two-dimensional also. So yes, even though they are two-dimensional, they are traditional ones, but they can be modified but for your purposes, and they have a huge role in the research activities. So if we find out the different types of assays carried out with the help of two-dimensional cell cultures in drug discovery and experimental pharmacology, there is a list of assays. You can find out that cell culture assays to determine the pharmacokinetics of drugs. For example, drug absorption, distribution, metabolism, and excretion. All these assays can be carried out with the help of two-dimensional cell cultures. Then cell culture assays to determine the drug toxicity, cell culture assays for preclinical drug testing, cell viability assays, drug permeability assays, for example, drug permeability through buccal, intestinal, nasal, ocular, rectal routes. And these are the specific routes which can be investigated for drug permeability as well as some drug delivery system permeability by using a specific cell lines. Drug screening assays, the major role plays by two-dimensional cell culture techniques is in the drug screening as a step of the drug discovery, where we use the high throughput screening like automated instruments, automated technology, where thousands to millions of molecules can be automatically screened on the particular assays, and that assays may be reporter gene assays, second messenger assays, cell proliferation cytotoxicity assays using the two-dimensional cell cultures. And definitely cell culture assay, two-dimensional cell culture assays can be used for research purposes to identify the mechanism of actions of the drugs. So these are the list of drugs which are popularly used, list of the assays which are popularly used for drug discovery and experimental pharmacology with the help of two-dimensional cell culture. I can't explain all the examples, but I can give you a popular example of two-dimensional cell culture, which is used in pharmaceutical industry for the study of absorption of drugs, and that is nothing but the uh, human colon carcinoma cells, CACO2 cell line. CACO2 cell line is actually form a tight junctions in a monolayer and mimic the intestinal epithelium. Additionally, CACO2 cells express the proteins that are involved in the drug transport. And that's why these unique properties of CACO2 cells making them a good model for testing the drug absorption. And that's why the CACO2 cell lines is very much popular in the pharmaceutical industry to find out the or to investigate the drug absorption in vitro or cell, with the help of cell culture. Similarly, MDCK MDR1 cell line is also used for such kind of drug transport analysis. So let us jump to the three-dimensional cell cultures. So definitely there should be some drawbacks in two-dimensional cell culture. That's why we are evolutionize the three-dimensional cell culture. Yes, two-dimensional cell culture has limitation of in vivo correlevance because they have less correlevance with the in vivo system, because inside our body cells grow in three-dimensional atmosphere. They don't grow in two-dimensional or one-dimensional, and that is the biggest disadvantage of two-dimensional cells, where we apply the force polarity. The cells grow in on a particular surface by adhering on that surface, and that's why they may lose their some original functions or lose them some original characteristics. And that's why to overcome these limitations, the scientists discover the three-dimensional cell culture technologies, where cells are allowed to grow and interact with the surrounding extracellular framework in three dimensions. So definitely three-dimensional cell culture refers to the culture of living cells inside the micro-assembled devices. So here some micro-assembled devices are used to support the cells, to provide the three-dimensional atmosphere to the cell, so that they can mimic these structures can mimic the tissue and organ specific microarchitecture, and cells can grow on that microarchitecture so that they can occupy the three-dimensional properties whatever they have inside the body. And that is the major advantage of these kind of cells. Another advantage is that due to three-dimensional culture, cells have the better cell-to-cell contact. They have intercellular signaling networks, and such growth of the cells or such three-dimensional culturing allows the cells to differentiate into more complex structures, just like spheroids or organoids, which can be very, very useful in drug research or other purposes. So majorly 3D cell cultures may be based on scaffolds or non-scaffolds. I will explain that in next slide. So if we talk specific applications of three-dimensional cell culture techniques in drug discovery and validation of drug screening, then definitely they have very huge applications. And from last decade, the use of three-dimensional cell culture in the drug discovery has been tremendously increased. And the reason behind is very simple, because in three dimensional, the cells retain organ specific functions better than the two-dimensional cell cultures, which often improves the in vitro in vivo correlation. So major drawback in the drug discovery is nothing but the in vitro in vivo correlation, and this can be improved by shifting from two-dimensional cell culture to three-dimensional cell culture, because 3D cell cultures and co-cultures have very close resemblance with the in vivo states, and they have the similar protein expression patterns, intracellular junctions. Practically no cell in our body grows in a monolayer. Actually, all the cells naturally exist in a complex 3D structures, and that's why three-dimensional cell cultures can give you the way by which the cells can interact with each other. Cells can interact with the extracellular matrix components, and that's why you can achieve, or you can acquire the original cell properties in your cell culture. Cells will have original functions, cells will retain their protein expressions, and that's why that may be more relevant to the original cells, and that will increase your in vitro in vivo correlation. So types of 3D cultures, yes, there are three major types of three-dimensional cell cultures. First is spheroids. Spheroid is nothing but the anchorage independent, that is non-scaffold based. In this technique, no scaffolds are used, only specific cell culture technologies are used to just form the spheroids. Spheroids are nothing but the cell aggregates. So these cells are cultured in aggregate form so that they can interact with each other, and they form the three-dimensional cell growth by techniques like hanging drop, low attachment plates, and magnetic levitation. Second is nothing but the cell culture scaffolds. So this is a scaffold-based three-dimensional cell cultures where they are also called as anchorage dependent. Here we provide some scaffolds. Scaffolds are nothing but the supports or mechanical supports to the cells on which these cells can adhere, and cells can proliferate and grow and migrate in three-dimensional way, because these scaffolds are designed or specially engineered in such a way that they can provide the three-dimensional microarchitecture structure to the cells. For this purpose, you can use the hydrogels, biological hydrogels like collagen, matrigel, gelatin, alginates, or sometimes you can use the synthetic hydrogels also, for example, polyethylene glycol, polylactic acid, polyglycolic acid, etc. So the scaffold-based three-dimensional structures are very popular in cell culture technology. If we talk about the third type of three-dimensional structure, is nothing but the specialized three-dimensional cell culture. They are of two types. First is microfluidic devices and micro pattern plates. Microfluidic devices are the cutting-edge technology recently discovered in the modern era of biotechnology, which will revolutionize the research field because of their great applications by relevance with the in vivo system. And that's why these microfluidic devices are nowadays very popular in the research field. So overall, if you think to carry out the three-dimensional cell culture in the laboratory, then you have to use these kinds of technique, which is called as first, it is liquid overlay, where low attachment plates like systems can be used. Then hanging drop technique, where the cell aggregates or spheroids are formed at the bottom of the tip of the drops. Then definitely hydrogel embedding, then spinner flask bioreactors. Then scaffolds are used. The scaffolds may be natural of origin or synthetic in origin. They provide the three-dimensional architecture to the cells to grow on that particular three-dimensional structure. And definitely, last is nothing but the three-dimensional bioprinting. This is the most advanced technique in the biotechnology where right now we are using. So if we talk about the microfluidic chambers, or these are also called as an organ on chip. So these are highly cutting edge technologies recently discovered for the purpose of research, and they have the great potential to be used in the drug discovery as well as experimental pharmacology. So how can you define these microfluidic devices or organ on chip? An organ on chip is a three-dimensional microfluidic cell culture chip that is engineered to mimic the physiology of an organ. So they are the engineered devices. They mimic the organ structure, and you can culture the cells in the three-dimensional way in that small microchip chambers. So obviously, these chambers are provided by the tiny channels where you can flow the liquids in the volume of microliters or picoliter volumes, and then you can transport the whatever the nutrients are required, and you can test your molecules in the form of chemicals or drugs on these cells. So These are not only under discovery, but many organs have been successfully transformed into the specific organ on chip. For example, skin organ, brain, eye, gut, kidney, heart, lungs, pancreas, bone, liver. All these organs have been successfully converted into the specific organ on chips. And here I have given the example of a breast cancer organ on chip. Here you can find out the in vivo arrangement of the breast cancer tumor, where you can find out that epithelial cell is containing the tumor cells, and the epithelial cells layer is also associated with the stromal cells. So this is a natural three-dimensional arrangement observed inside our body whenever we suffering from the cancer. And similar arrangement can be done in these chambers of the chip, where you can find out that this is the mammary epithelial layer, then this is the spheroid of ductal carcinoma in situ, that is cancer cells forming the spheroids. This epithelial layer cell is also having the extracellular membrane, then stroma is also there, and you have the fibroblast in connection with this cell. So all the in vivo structure resemblance can be obtained here in the chip with the help of three-dimensional cell culture. And that's why these organ on chips are referred as a cutting-edge technologies, having the interdisciplinary applications for disease modeling, drug testing, precision medicines, and they will definitely revolutionize the drug development pipeline soon. So these not on these organ on chips are not only under research or under development process. Already there have been some well established organ on chips available, and some pharmaceutical industries or some research institutes already they are using that. And if we talk about their applications in the drug discovery platform, so definitely there are PDAC on a chip already established and used for early drug discovery in drug discovery pipeline. For basic research, similarly, glioblastoma on a chip, that is brain cancer on a chip. This chip is also used for a drug discovery pipeline. NSLC, that non-small lung cancer on a chip, this model is also been used for drug discovery stages. For preclinical stages of the drug discovery, human on a chip is interesting modification of the organ on a chip it has been also developed, and where the different chambers of the chip acts as the different organs. First pass multi-organ chip has also been developed for predicting the pharmacokinetic values of nicotine. For preclinical trials and translation, again, liver lobule on a chip that is has been developed for drug-drug interactions. Multi-sensor integrated organ on chips have been developed for these preclinical trials and translations. That means already these microfluidic devices, that is organ on chips or human on chips, cutting-edge technologies have been reached to the drug discovery processes. And yes, no doubt they are in early development stages, and they are very costly. It will take some time to reach these kinds of technologies to the regular use of pharmacological laboratories. So overall, if we summarize the applications of three-dimensional structure for drug discovery and pharmacological experimentation, then you will find that yes, target identification stage, high throughput screening of drugs, drug molecules under investigation, lead optimization, preclinical, and up to the clinical trials. These three-dimensional cell culture technologies can play important role in the form of spheroid cultures, organoids cultures, and organ on chips. And that's why I said that two-dimensional cell culture as well as three-dimensional cell culture has tremendously entered into the drug discovery pipelines and experimental pharmacology principles, so that they are revolutionizing the drug discovery process. So yes, cell culture evolution actually carrying out the revolution for drug discovery. We have moved from two-dimensional to three-dimensional, now three-dimensional to the organ on chip, and we have designed some specific organisms or animals also with the help of these cells, like cancer xenografts or zebrafish models, where we can use the cells along with the animals to design the specific animal models. Human on chip is also one great advancement, where you can convert this chip into a different parts of into human body, where the different chambers of the chip will act as a different organs, where you can culture the cells of that specific organs in three-dimensional way. And that's why the cell culture evolution is revolutionizing the drug discovery. So if we talk about the major breakthrough discoveries carried out in last decade, specifically Nobel Prize winning discoveries, where the cell culture technologies play an important role during the discoveries also and applications of these discoveries also. So one of the great example is nothing but the Nobel Prize for 2020 given for chemistry to the Emmanuel Carpenter and Jennifer Edonda for their discovery of CRISPR technology, which involves a wide, huge use of cell culture technology. Second example is nothing but the Nobel Prize for Physiology or Medicine in 2019, where that was given to the William Kelly, Peter J. Ratcliffe, and Greg L. Semenza for their discovery to identify the how cells adapt when oxygen levels drop. So this discovery also awarded the Nobel Prize in 2019, which was based on the cell culture techniques. Again, the Nobel Prize of 2018 was awarded for physiology and medicine was awarded to the James P. Allison and Tosuko Honjo for identifying the two highly specific drug targets that are PD-1 and CTLA-4. And these are the two actual proteins which are actually inhibiting the immune cells from attacking the cancer cells. And this is a major breakthrough in the cancer drug discovery, because due to this discovery, now it is possible to cure the stage 4 patients also, particularly melanoma and related cancers, due to these anti-PD-1 and anti-CTL4 drugs. And that's why these kinds of research, which are based on the cutting-edge cell culture technologies, were awarded to the Nobel Prizes. Again, 2016 Medicine Physiology Nobel Prize was awarded for the research of autophagy to the Yoshinori Oshumi. So yes, these are the potential applications of cell culture technology for drug discovery and experimental pharmacology. So I think pharmacy students should take this seriously, and they have to develop their skill sets, and they have to develop themselves for such kind of opportunities, because this field is having great opportunities for career, and you can become a drug discovery scientist, as well as you can develop discover some new drugs, and there are tremendous opportunities in this field in the coming future. So I hope this lecture will motivate the students for this field, and pharmacology experimentation will also will improve with the help of these cutting edge technology like cell culture technology. So thank you. I'm ready to take any questions.
It's a really interesting and knowledgeable session given by you, sir. So, sir was focused on especially 2D, 3D, human chip, and cell culture evolution. So, without any ado, I would like to choose a few questions from chat box. Sir, there is a question from Professor Rushikesh Velis. He want to ask that what are the latest development in the area of genotoxicity or mutagenicity testing apart from AIMS test?
Yes, yes. So mutagenicity and testing have been revolutionized. Sorry, they have been also using now the three-dimensional cell cultures, and many assays are under development. But yes, there are specific assays used for identifying the mutagenicity and other purposes. But here also important to understand the molecular biology applications are also used along with the cell culture. So that's why the genomic advancements are also used for advancement of these mutagenic assays. I can't tell you the specific assays nowadays you are using in pharmaceutical industry, but definitely the three-dimensional cell cultures are playing important role in this advancement of assays.
Yes, sir. Again, there is a one question from chat box by Tanmay Patwardhan. He want to ask that, are there any way to reduce failure rate of drug molecule going to clinical trial, especially drug from psychotic disorders?
Yes, definitely. Any type of drug is there, not only the psychotic disorder drugs, but definitely all types of drugs have the problems in the drug discovery process when they fail, and that problems may be the a major problem is nothing but the target identification. So, to any type of disease, it is important to understand the disease biology first at a molecular and cellular level. If the disease biology research and early target identification research was carried out in a specific manner, by using a cutting edge technology like cell culture technology, where you can use the human cell lines, because understand that human cell lines are from humans, and that's why they have a very high relevance with your human body. And if you use the three-dimensional cultures, they provide again more relevance to the in vivo cell attachment or cell growth. And that's why such kind of experimentation, by modifying your experimentation from two-dimensional to three-dimensional technologies, similarly identify the targets by detailed studies on the molecular levels as well as cellular levels, because many times poorly targets are identified. Most of the times the focus will be given to the receptors, but I just told you that many times there are some proteins which are involved in the disease biology. They are beyond the receptors, and definitely many times only receptors are not playing important role in the disease biology. There are many proteins which can play important role, just like I give you the examples of PD-1 and CTL4. These two proteins not actually the receptors, but they are involved in the inhibition of immune cells. So yes, by just carrying out the detailed studies in the drug discovery early stages, you can minimize the failure. The specific pharmaceutical mantra is what? Fail early, fail fast. So this is a mantra followed by pharmaceutical industry. What this means? This means that we have seen that there are 8 to 10 different stages of drug discovery. So if your drug fails in a clinical trial, there will be tremendous loss of money, tremendous loss of energy, tremendous loss of manpower, tremendous loss of time. So pharmaceutical industry wants, if the particular molecule is going to fail, that should get failed in early stages, so that we can minimize the loss of money, we can minimize the loss of time. And that's why to fail early, fail fast is the mantra of pharmaceutical industry. And to avoid that, or to achieve that, you have to carry out the initial drug discovery very seriously, and that should include cell culture technology like approaches, so that you can increase the drug discovery success.
Yes, sir. And last question from, I think, from degree student, how to initiate the drug discovery at college level?
Yes, definitely. Very interesting question, and that is the purpose I given this lecture, because unfortunately, pharma students forgot that they can discover the drugs. Mostly, pharma students are focusing on the careers where they can formulate the dosage forms, or they can make the careers in the industries, in quality control departments, and like. But they forgotten that they are the original people who can discover the drugs, because they have all the knowledge regarding the drugs during their education. And that's why to initiate the process of drug discovery in the college laboratories, I told you that for that purpose, college and university level involvement is also required. Management also sets some setups like cell culture biology and molecular biology labs in the pharmacy colleges, so that you can carry out some early stage drug discovery research activities in your college, just like target identification, different cell culture assays to identify the pharmacological effects or investigate the mechanism of actions of drugs, natural molecules on human cell cultures. These kind of study, you may find a unique molecule which is not reported in your college also. I can give you example from Pune itself. The One ISRA is the research institute in Pune, led by the Dr. Subhash Padde. He is a director of that ISRA institute, and Alana College campus of pharmacy. They collaboratively do the research, and along with their US collaborators, they just identify the curcumin derivative in their laboratory, and they tested this into the cell cultures, and they design the curcumin leads and hits to leads in the America, and they just investigate that molecule in the cell culture, and now they got the US patent for this molecule against anti-breast cancer, and that's it. That is a huge thing. So these kinds of studies can also be easily carried out in the colleges, but for that purpose you need some cell biology and molecular biology setups.
So definitely after this question answer session, I hope number of points will be clear by of the student as well as all the different types of staff member who present in this seminar. So after that, I just say thank to respected Dr. Aniket Mari sir for this knowledgeable session, and I hand over this session to Shubham Khairnar sir to continue this program. Thank you, sir.
Thank you, sir. Thank you very much.
Okay. Good morning, respected dignitaries and delegates out there. I, Professor Shubham Khainar, working as Assistant Professor in Department of Pharmacology, MET IIP. Heartily welcome all the delegates and our next eminent speaker. I am extremely privileged to introduce Professor Sadhna Sate. Ma'am is working as a professor at Institute of Chemical Technology, Mumbai. Uh, Dr. Sadhna Sate, ma'am, is an eminent research scientist working in the field of pharmacology and toxicology with an academic experience of 30 years. Overreaching field of her research encompass various neurological or neurodegenerative disorders like epilepsy, Parkinson's disease, Alzheimer's disease, as well as metabolic disorders like diabetes mellitus and related complications such as diabetic neuropathy. The MAM's overall research focuses on establishing novel therapeutic interventions by exploring herbs and modern medicinal drugs. MAM is really a great research also. Sate ma'am successfully guided 45 PG and 16 doctoral students and currently supervising 15 masters and 5 doctoral members. Ma'am also have a number of professional affiliations such as nominee of CPCSC committee, chairperson of Institutional Animal Ethical Committee, then life member of University Department of Chemical Technology Alumina Association, and life member of Society of Toxicology and life member of Indian Women Scientists Association. Ma'am have number of international and national research collaborations also. She has over 80 national and international publications to her credentials and has been renowned speaker in several esteemed and national and international conferences. Her horizon is not limited to academia, but also as a consultant to the global pharmaceutical industry, where she extends her services in regulatory toxicity studies and pharmacodynamic evolution. This is just a brief introduction of this human personality, that is Dr. Sadhna Sate ma'am. Thank you, ma'am, for joining us at this national webinar. So I request respected Dr. Sate ma'am to share her valuable knowledge and guide us all. From here, you can take over the session, ma'am. Now,
Yes, yes.
first, ma'am, you can proceed for your session.
I'll just share my screen
Yes, ma'am. Yes,
first to begin with, and then we can talk.
yeah, ma'am.
Mute yourself.
One sec. I'm not able to. I mean, yeah, I couldn't see that. One sec. We tried yesterday and we could do that. Somehow that goes off when I.
Okay, now it is there, madam.
It is there. Okay.
Yes, madam.
That's nice. Yeah. Okay, that's the window and
No, hold on,
share.
madam.
No, entire screen, should I?
Yes, madam.
Okay, 1 minute. It just goes off, the minute I change the window, it just goes off.
Could you please try one more time, ma'am?
Yes, I'm trying. This is what I'm trying right away. See, the minute I try and join you guys. Okay, let me just go back and let me just do it once again.
Yes, ma'am.
It's got hanged. Can I go out and come back again? I think that should be fine.
Sure, yes, madam. Yes, madam. Sure. Go ahead.
Wait. It's doing something. I can see the thing. It never happened yesterday. It is happening today.
Yes.
Anyway, thank you so much, everyone, for kind introduction. And I'm very happy when Dr. Pawan told me that, Madam, just talk to our students, the masters as well as Dr. Pawan. Even bachelor students have joined today.
No, madam, faculty members and PG students have joined.
Oh, okay, okay. So welcome all faculty members. Just hold on with me because the Chrome is not responding.
Madam, we can see you.
Yeah, yeah. I'm just trying to share the presentation now.
Yes, madam.
That windows only not being seen. The the
If it is
presentation
not,
window.
can you share that? Have you shared that?
When I try doing that now, like I open the window and when
Okay.
try doing share it, it just goes away from there. Now it's there. It goes within 2 minutes.
Uh, if that persists,
Can
the
I
problem
just
is
send you the? Can I
email.
send you
Yes.
the presentation?
Yes, madam.
I'll do it
Please.
right away. Yeah.
Yes.
I'll try only once if possible. Otherwise, I'll send it to. Anyway, not a big deal. See, you must be able to. It goes. I'll send it to you. One sec. Huh?
Madam, have you sent that email?
Yeah. I'm just give me your email ID, fatafat,
It is
and I'll
Pavan
just
Udavant. Yes,
yeah.
Pavanudavant
Huh. Hmm.
@gmail.com.
Okay. pavanudavant@gmail.com.
You can see it on the
Just
screen as well.
yeah.
Yes.
Mm-hmm. Just check whether you've got it.
Yes, madam.
You've got it.
No, madam, not yet.
Okay. You will get it.
Yes, madam, got it.
Huh? So can you share that?
Yes, yes. In a minute. I'll-
Share screen. Share screen. हो इथून करते.
Okay, madam, can you see the screen?
One second. Yeah, I can see the screen. Yes. Uh-huh. I can see. Yes.
Madam, please, you
I'm
can please
yeah.
proceed. Yes.
Yes. And Dr. Pawan, will you change that? I mean, the
Yes,
slides
yes,
for
yes,
me.
for sure. For sure.
Okay. But it could have been better if I had it with me.
Yes, it
So
will take 1 more minute. It will take 1 more minute. We are arranging for the same.
okay. So I'll start with it. The first slide. Can you just go to the first slide before this? Let me express my gratitude towards you. The first slide. Yes. So, I thank Dr. Pawan and Dr. Neelam, who are always in touch with me for this particular lecture. And they wanted the faculty as well as the postgraduate students to know about the newer trends in experimental pharmacology. I have been visiting your esteemed institute as a CPC SCA nominee, maybe for last 2 3 years now, and that's when while we used to consider your protocols, I used to really take interest and talk about how a particular study could have been done better, or what are the new rules and regulations, and particularly the new trends in experimental pharmacology that we have to talk about is simply because of very strict regulations by the CPC-SCA in the use of animals. And that's the reason what we feel, meaning even at ICT, that the master students can actually perform the in vitro experiments before they could use the animals for that matter, because uses of animals is extremely difficult, rather regulated in today's scenario. But then you can't ignore the importance of animal studies, because you don't get a 3D system like that of animals, which is very close to human beings. So it's very important to have uh, basic knowledge that yes, animals are the final model that we could use to have many experiments in pharmacology, or to explore many activities, biological activities in pharmacology. But before going to animals, there are simpler experiments also which you can do, and you can go towards those animal experiments in a better manner. So that's the reason today's lecture, that is new trends in experimental pharmacology. I'm very happy that the lecturer before me, Dr. Mali, he explained about the 3R principles and how the animal usage is difficult. And then how cells could be used in evaluating or exploring biological activities. Now we will see other aspect of this experimental pharmacology. Can you go to the next slide, please? Or can I do it on my own? Okay.
No,
So
madam. No, madam. You won't be able to do that.
no problem. You do it.
I'll do it.
Yeah.
My pleasure.
So basically, understand everyone. Research methodology is to investigate the study of material and sources to establish facts and have new conclusions. And you have a peculiar methodology for that particular matter. So research methodology is very important. Dr. Pawan, next slide. Next slide, please. Okay. So, I have animated it. So you go on clicking on 111 and you can click on all the steps. This is you meaning yourself, the experimenter or the research scientist. And Dr. Pawan, you could just, uh, go on clicking to see all the steps. Yeah. So define a problem, what you want to study. Define that first. Dr. Pawan, you can, yeah, just go on clicking all of them so that yeah. Mm-hmm. Fine. So remember, no, go back. I'm sorry for this. Dr. Pawan, can I try it once? If I could do it, hold it, try it from my side. You
You
already
can,
have it. Yeah,
yes,
if
yes.
I
You can
can
try it
try.
once. You can try it
Huh.
once
I'll just try it once because, I'm not so happy with this.
because we could do it at our end. So you two could should be
Yes.
able to do that.
Yeah. I feel so. Let me try.
Yes.
No, not possible for me. I don't know what's wrong. Should I just change over to another this thing, internet, and let's see if I could do that. Just give me a minute, and I'll try with another internet. That's what we discussed yesterday.
Okay, madam. Okay.
Okay, I got it. I was wrong. Thank you. Anyway, can you see my screen now, everyone?
Yes, madam.
Hello.
I can see the
Ah,
screen. Okay.
thank God. Now everything is in my hand. I'm happy about it.
Yes, yes. You can control it now.
Okay.
Yes,
So
madam.
thank you so much. Okay. So we were understanding about the newer trends and research methodology and the 6 steps that you need to achieve your goal. So basically, you define a problem, what you want to study first. Do a thorough literature survey. Trust me, we all are really lucky in this era when we have Google in our hands. Google and of course Google Scholar. So once you see how others have studied a particular problem which is similar to your problem, then you can design your study. And once you design, you can even execute it. You can apply statistical significance because statistical analysis also happens to be one of the most important things. And remember that without publication and patenting, no one will notice you. So to be noticed by everyone, it is very important that you must have, you must go to the last stage, that is publication or patent. Don't just stop at doing the study and okay, I couldn't publish it. No, try publishing it as well. Now, simple thing, pharmacology is so interesting that you have these various branches in which you can work. So you have molecular pharmacology, biochemical pharmacology, literally enzyme assays and all will come here. Receptor assays will come in this molecular pharmacology. Pharmacokinetics and drug metabolism. Supposing your colleague produces or formulates a particular new formulation for a particular drug already existing, then you could just carry out pharmacokinetics and its distribution and its metabolism. These studies also are considered as pharmacological studies, and without these studies, your colleague's formulation won't be considered as a standard formulation or a good formulation for that matter. Everything needs to be verified using toxicological studies. And I think the next speaker will definitely talk about the different toxicological studies, even chemotherapies, meaning not only cancer chemotherapies, but even antibiotics, antibacterial evaluations, they will come in this. And systems pharmacology is what generally all of us think of pharmacology is. So pharmacology is not only systems pharmacology, but trust me, it encompasses all these things. So see, you as a pharmacologist, you have so many avenues that you could explore. I'm just enlisting, you all know this. I'm just enlisting a few of them, like neuropharmacology, like Dr. Professor Subaidar, he mentioned that his area is neuropharmacology. I also, to certain extent, ponder in this neuropharmacology, cardiopharmacology, gastrointestinal, meaning acidity, what do you say, spasmodics, antispasmodics, all those, that is gastrointestinal pharmacology, respiratory pharmacology, and immunopharmacology. Now, this particular branch is gaining much, much importance these days due to COVID. Now, what could be your methodology to do these experiments? It could be actual animal experiments, that is, we call it as in vivo, or it could be in vitro assays, or it could be in silico assays. Trust me, in silico and in vitro, if you concentrate more, your design of the in vivo studies will be perfect and absolute. Now, I'm just taking you through different kinds of in vitro and ex vivo assays. I'm just trying to show you that you can, do certain antioxidant assays, which is also a part of pharmacological assays, okay? Because antioxidant assays give you a clue about different activities, biological activities of a particular substance. Or it could be enzymatic assays. So we are going to discuss about this in great details and how you could do that. I'm also going to ponder about these because these two I feel are really simpler assays as far as you can get it done with your master students. Then drug permeability assays. So drug permeability, see, why do our colleagues make newer formulations? They make newer formulations so that the drug is bioavailable in a smaller dose. So how to confirm that? So you could do these drug permeability assays. You have in vitro procedures as well as in vivo procedures for that matter. Then drug transporter assays. So there are many transporters which, like we all know about the PGP or breast cancer resistant protein, which are efflux protein. So you could actually work out to check if your product is inhibiting these transporters so that it could be better permeable or better biodistributed for that matter. Or you could have cell and organ based assays. So cell and organ based assays meaning using cells, as Dr. Mari told you, and because you know what kind of cells you're using and what is your end point, you can design a study. We will discuss in QA session about this if you're interested. Cytotoxicity assays, I guess Dr. Mari has discussed that. Cellular and molecular biomarkers, these are, you know, when you do cell and organ based assays, you can literally have these cellular and molecular biomarkers, something like um, a brain derived neurotropic factor, which you can study it by growing the neuronal cell lines. Or by growing hepatocytes, you can check SGOT and SGPT. These are the cellular biomarkers for that matter. So, you know, biomarker study could be also undertaken, and bioassay. In BPharm, I think we expose the students to some extent to these bioassays, but this could be taken ahead as a major in vitro or ex vivo assay for that matter. So now, once you do the biological activity, it is very important to show a pharmacokinetic and pharmacodynamic correlation. So I'm just trying to tell you that as a pharmacologist, using animals, you can do this. But this is in vivo, so I'm not touching it. And pharmacodynamic studies, particularly meaning biological activity studies, they particularly help you in studying the action of a drug in the body, measuring the effectiveness or efficacy of the drug, and by doing toxicity, you can also do benefit risk ratio and understanding how and why a drug works. See, whether the drug works or not, yes, that is efficacy. But how it works, why it works, the mechanisms, if you could explore, that would be ideal to endorse any pharmacodynamic activity. And of course, PK and PD correlation has to be there. So, in pharmacodynamic study, concentration response relationship is very important, which could be of two types. One is quantal, that is all or none response. So either you have seizures by prevention of seizures by phenytoin, or you have graded response where increasing the concentration of the drug, if you could increase the uh, if efficacy. Just give me 1 sec. Huh? Just give me 1 sec. One sec. Huh? Yeah. So we were here and we were discussing about pharmacodynamic studies, and we were discussing about graded response where effect increases with concentration. Okay, now PKPD modeling, as I said, it's important whenever you do any pharmacological study so that the compound should reach the target. So PK studies, compound should bind to the target and show its biological activity. PD studies, and even exploratory receptor binding, etc. And compound should have a functional modulatory effect on the target. So you know it's very important to have a PK/PD correlation for that matter. And there are different softwares available. Once you get the data, you can always put it in that, and you can study that. Toxicological studies, you can do that to study the adverse effects of chemical, physical, or biological agents on people, animal, and environment. And why do you do toxicological studies? Because you need to prove the safety of a particular preparation or a phytoconstituent or a what you say new chemical entity for that matter, or a new formulation. before administering it first to the human beings. So there are different regulatory guidelines through which you can do these toxicity studies. And I've just listed a few of them. ICH is also there. And what you could study the toxicity of, you could study the toxicity of drugs or generics or NCEs or chemicals from various sources. It could be pollutants, anything. Meaning, as a pharmacologist, you can think of so many things. Drug delivery systems, your colleagues who are in formulation, pharmacognosis to pick out phytoconstituents, or even Ayurvedic preparations. You could study the toxicity of this as a part of pharmacology. And in vitro, you could do it in cell line assays, or you could study dermal irritation, ocular irritation, or biocompatibility of polymers. You know, something like when people ask you, like you wear lenses inside the eyes, not the contact lenses that we wear, but the lenses that are put as a part of cataract and cataract removal. So whether they are biocompatible, I'm just talking of one polymer, but there could be so many polymers which you use in formulation. You could check their toxicity study as a part of pharmacology. And of course, in vivo, there are many like acute, subacute, chronic, dermal irritation, ocular irritation, all these using animals and all these using cells for that matter. And all these toxicity studies ensure safety, and this is also a part of pharmacological studies. Now, okay, now let's come to the main problem today, meaning my major content for today, like how to arrive at a research problem. So, I my area of expertise is neurodegenerative disorders, particularly Parkinson's, Alzheimer's, and neurological disorders like epilepsy, and of course, metabolic disorder like diabetes. And trust me, diabetes also affects all these. Uh, uh, what do you say? Mm, disorders as well, meaning there is a correlation between diabetes and these neurological disorders. So when I say, okay, I want to do a research on diabetes, it could be your choice. You want to do it on hepatoprotectase, you want to do it on kidney protectase, it's your choice. But then how to arrive at a research problem. So first you find out before choosing a problem, what is the statistics and what is the prevalence, whether it is really worth looking into this problem. And trust me, even if prevalence is not much, and if the disease is rare, and if you wish to explore it, please go ahead. I'm just trying to say that generally when people do, people look into the worldwide prevalence, the pathophysiology of type 1 and type 2 diabetes, and then associated complications like nephropathy, neuropathy, retinopathy, cataractogenesis, peripheral vascular disorder. I mean, what I'm trying to say is you could have so many sub problems for you to study it. Now, we have been working on diabetes and related complications for a long time. And let me just take you through one simple slide wherein hyperglycemia, meaning increased blood sugar levels, you all know that it would increase intracellular glucose level. Now, where intracellular glucose level only in those organs which pick up glucose independent of insulin. So, which are these organs? Heart, liver, brain, kidney. Because they are the ones, they have to have energy all the time. So, they have to have glucose all the time. And so, these are the organs which actually get affected when diabetes sets in. So intracellular glucose levels increase inside the cell. So glucose gets, phosphorylated, and it just gets, into that TCA cycle, and it gets into the energy production cycle in the mitochondria, and it generates a whole lot of reactive radicals. When high levels of glucose are there, lot of reactive oxygen species are generated, and these reactive oxygen species literally would produce dysfunction of the mitochondria. So when mitochondrial dysfunction is there, you have so many different species of uh, ROSs. So you have oxygen, but superoxide, peroxide, hydrogen peroxide, hydroxyl, and hydroxyl radical, iron and radical. So you have so many ROSs, and these ROSs lead to activation of different biochemical pathways, resulting in oxidative stress. So what are these pathways? Polyol pathway, AGEs, that is advanced glycation end products, protein kinase C, hexosamine pathway, etc. So when all these biochemical pathways are activated, and you have the diabetic vascular complications, micro as well as macrovascular complications. So what would you feel? You would feel that if you stop sugar, if you control the sugar, will all these will be stopped? Will they be stopped? That's the question. And trust me, the answer is no. Answer is no, simply because when there is a mitochondrial dysfunction, there is a metabolic memory. Mitochondria are capable of remembering that there is something wrong in the oxidative, I mean, too many oxygen, reactive oxygen species being there. And that's the reason it it gets into a different mode of producing more and more ROSs. So what should you do that? So what would actually work? It's not the real control of sugar, but if that is anti-diabetic, yes, that would be number one kind of a treatment modality. But antioxidant and anti-inflammatory also would be one of the major things or major pathways that you could consider. And that's the reason I give much more importance to antioxidant as well as anti-inflammatory levels as well. Okay. Now let us see. Now you just checked four pathways here. Let me just take you through one pathway, that is protein glycation pathway, that is for age formation, advanced glycation end products. So what do you have? This is a protein with an amino group, and the glucose, if extra glucose is there, then it, it produces Maillard reaction, and it produces Schiff's base that produces amidori products, and within weeks it produces advanced glycation end products. And not only it stops at that, but proteins like albumin, which is in huge amount in the blood, the ages that are formed from combination of glucose and protein, they go and attach, and the proteins get glycated. The proteins get glycated. We generally measure glycated hemoglobin in the blood as a what you say severity of the diabetes for that matter. So okay, amidori products are particularly alpha dicarbonyl compounds like glyoxal, methyl glyoxal, and 3-deoxyglucosone. And this also, meaning amidori products, when they are formed, there are ROSs generated. Ages, when they are formed, ROSs generated. Alpha carbonyl compounds, alpha dicarbonyl compounds, they get rearranged to form Schiff's base. Again, ROSs are produced. So you can see this is called as carbonyl stress. But you can see everywhere reactive oxygen species are produced, everywhere. And this is what is dangerous, because once these are produced, it starts towards the inflammatory reactions, and it starts towards cellular degeneration for that matter. So we could easily control these things, and we could have we could perform our experiments in that sense. So what in vitro experiments that we could do is, one is I'm just talking about the last two slides that I explained, that uh, age, mitochondrial dysfunction could happen, and because of that ROSs could be generated. and because of that many pathways could be activated leading to inflammation. And then I just discussed about one pathway, that is age pathway. So let us consider antioxidant activity. Very simple experiment, total phenolic content and DPPH assay. Now why I'm showing you this EASA? So EASA is essentially ethyl acetate extract of Saraca indica. So ethyl acetate extract of Saraca indica is what I'm trying to explain you. And let us go here. Oh, I can't. 1 minute. Let me go back to my presentation. Yeah, can you see that now? Hello?
Madam, we can see the screen.
Okay. But you can see the screen, right? The total phenolic content of
Yes.
EASA.
Yes, madam.
Okay, so
DPPHS.
simple antioxidant activity. I have showed you about EASA. It could be of any reagent, anything, any compound. It could be an NCE. It could be a phytoconstituent. It could be an extract. I'm showing you activity of extract itself. So phenolics, they include phenols, phenolic acids, coumarins, flavonoids, stilbins, hydrolyzable and condensed tannins, lignans, and lignin, meaning all these are plant products. So, whatever plant products that you wish to study, you could actually study for total phenolic activity. And remember one thing, phenolics are responsible for antioxidant activity. They contribute a lot towards and antioxidant activity and protective agents against even UV light. Meaning, if you want to make any cosmetic to protect from UV, you could do this total phenolic content of that particular substance for that matter. Okay, although quantitative determination of polyphenols is hampered by structural complexity, various methods are there to uh, evaluate the phenolic content. So polyphenols in plant extracts, they react with special redox reagents like fallen CO2 reagent, and they form a blue complex which can be quantified by simple visible colorimetry. So the reaction forms a blue chromophore with fallen CO2 reagent by tungstic phosphomolybdenum complex, and all this is available in all literature, meaning research papers. So I'm not showing you anything great. I'm just trying to show you that you could do total phenolic content as a matter of its antioxidant activity of any compound. So higher is the absorbance at this 765, higher will be the total phenolic content and higher will be the antioxidant activity. So we chose standard gallic acid because some standard you have to use. So the gallic acid is simple to analyze. You can also use that, and you should just do a concentration versus absorbance curve. And always remember that your method should be so good that you should get this R square almost equal to 0.99, meaning that's the best. But 0.96, more than 0.95, even more than 0.93 is also okay. So average absorbance of my extract EASA was 0.754. So I just put that in this reaction and I get 45.79 micrograms of gallic acid equivalent, meaning the extract contains so many micrograms of gallic acid equivalents in it. So that's meaning it is compared to gallic acid for that matter. So you can decide of different extracts which one is better. So higher the uh, value here, better will be your antioxidant activity for that matter. Give me 1 minute. I'm sorry for that. Okay. Hmm. So the next antioxidant assay is DPPH assay. Now this is DPPH is another compound. It's a radical. It is considered as a radical. So the radical scavenging activity of this. I request you just give me 1 minute. I'm really feeling
No,
meaning
madam, it's
the
absolutely fine.
throat
Yes,
is
you
getting irritated. Just give me a minute.
you take your time, madam. It's absolutely fine.
Yeah. So another assay that we can have a look at is the DPPH assay, and DPPH
Madam,
is a rad. Yeah. Can
excuse
you
me, madam.
hear? Yeah.
Yes,
Yeah.
we can hear you. Could you please put your presentation on the slideshow mode, madam?
Okay. Sure. Sure. I'll do that. Sorry for that. Yeah. Can you read that now?
No, it is still not. It's not.
Visible now?
It is not visible, madam. It is
Okay.
not being.
Yeah. 1 minute. I'll try again.
It is not on full screen.
Yeah. 1 second. Check now.
No, not yet, madam. Maybe
See.
you can do it from the top control panel.
Okay. I'll try doing that. Possible?
No, not yet, madam.
Okay. I'll come out of it and do it again.
No, madam, you can continue. It's okay. It's okay, madam. You can continue.
And the presentation is with you. So, uh, I'll try, of course, I'll try.
No, madam, you can continue. It seems there is a problem, but you can continue anyway.
Yeah. Can you see it now anyway? Because,
Yes, madam. Yes. Please go ahead.
okay. So, DPPH is a radical, and you can actually, a very simple method. The sample whose DPPH scavenging activity that you want to measure, you can dissolve it in methanol. And always important is that you have to use various concentrations to check any activity. That's very important. And these are added to 100 microliters of DPPH. So the capacity of a substance or an extract or a chemical to scavenge this DPPH will be seen in this assay. So DPPH solution was freshly prepared under dark conditions, and equal amounts of methanol is added to the control. The samples are incubated. I've written where because I'm just showing you my experiment. You can work out with your experiment, and you can incubate it for 20 minutes and read the absorbance at 517 nanometers. The method is optimized using ascorbic acid as a standard. The way we used gallic acid as in polyphenolics, we use ascorbic acid in DPPH scavenging activity. And here you actually get to understand the IC50 or inhibition by the compound. So IC50 of the ascorbic acid, meaning vitamin C, is 4.5 microgram per ml, whereas IC50 of ESA meaning my extract, is 28.14 microgram per ml. IC50 of what? Inhibition of DPPH radical. So that means naturally ascorbic acid is more powerful because it is inhibiting 50% of DPPH at 4.5 micrograms per ml, whereas my extract is inhibiting it at 28.14 micrograms, meaning extract is not as powerful. But understand one thing, you can do many more experiments in this to find out the IC50, and one more assay you can do as antioxidant activity. Uh, so this is just about IC50 values and its interpretation. You can read it. I've shared the presentation with Dr. Pawan. The next experiment, this is also very, very interesting. We say that ages, meaning advanced glycation end products, they particularly uh, produce all those deleterious effects like inflammation, etc. So we can even do this in vitro experiment to see whether a particular substance can inhibit the advanced glycation end product for that matter. So what you do is very, very simple experiment. You have bovine serum albumin, you get it in the market. All you do is you take 10 milligram per ml of bovine serum albumin in an Eppendorf tube, add glucose solution at 5.5 molar concentration, and the what you say one, you keep blank control with BSA and PBS, meaning no glucose added here, meaning no ages will be formed here. Ages will be formed here. And you have one standard control. What is that standard control? That is aminoguanidine. So aminoguanidine is a substance which inhibits the advanced glycation end product formation. Okay. So what you do is you add bovine serum albumin and glucose in the same concentration as the age control. Plus you add aminoguanidine as 4.5 micromolar. So you have this standard control, you can say positive control, the one which inhibits the ages. And then you can have your test compounds. Now, in this study, I have used gentisic acid and ethyl ferulate because we were exploring these two substances for its anti-age activity. So now you can understand. You can literally take any substance which you feel could have anti-age activity, or like supposing if you know from traditional literature, traditional research or traditional therapeutics that a particular herb works, find out all the phytoconstituents in that and you could just choose few of them, or you could check the activity of compounds which are synthesized by your colleagues. You could just do anything. Only thing, remember one thing, you should have graded concentration. And this graded concentration you will come to know by, optimizing, meaning you will have to little work out on it. This is after working out, optimizing the experiments. But this can be a full-fledged informed project also. That's what I'm trying to tell you. What you do is you just incubate it for 14 days, all these things in an incubator at 37 degrees centigrade. And there are different assay procedures. I'm not saying something new. All this is available in the literature. So you have how to, you can actually go to the end product, that is age, or you can find out how many amidori products have been formed, or you can find out the thiol content or protein carbonyl content. So, remember one thing, carbonyl, amidori, and age formation, these are the deleterious products of ages. And thiol is a good substance, meaning it's a protective substance. So, your chemical or your phytoconstituent should, meaning ideally, yeah, not should, ideally, I mean, you expected to increase the thiol, but it expected to really decrease all the others for that matter. Have you understood this? Meaning, I hope I'm not confused you. It's a simple experiment wherein you just put BSA plus glucose plus your test compounds in various concentrations and simply incubate. At the end of 14 days, there are various procedures to find out ages, amidori, thiol, protein carbonyl content, and you can do that. Okay. Now, for gentisic acid, if you could see, you get a dose related inhibition of age formation. Increase the concentration of gentisic acid, and you get a dose related increase in the inhibition of age, meaning gentisic acid can inhibit age formation. And aminoguanidine, if you can see, 10 micromolar has almost equal to aminoguanidine standard concentration at millimolar. Aminoguanidine is millimolar, and you have micromolar concentration equivalent to aminoguanidine. So, do you see one thing that our herbs, they have a great potential, really, and that has to be looked into. Meaning, we should not stop ourselves. We should explore the herbs for that matter in such simple experiments. Now, ethyl ferulate, if you see, haphazard results are being seen. You don't see a nice dose related response. It increases, then again it decreases. After increasing the concentration, in a percent inhibition decreases. It's not consistent. And why that was so? Because this particular substance was not soluble at all. And if you see the controls and everything, they are all water based, meaning aqueous. And that's what happened. Because of its poor aqueous solubility, ethyl ferulate could not show it. So now we are taking this ahead by solubilizing ethyl ferulate. So when you solubilize ethyl ferulate, naturally whatever you use to solubilize it, that also has to be taken as a control in your experiment. So I just want to show you that ethyl ferulate and gentisic acid, just giving you two examples of in vitro inhibition of age formation. Now next is enzyme inhibition assays. Again, very, very simple. If you must have studied as a pharmacologist that there is something called as a carbose, which inhibits alpha glucosidase enzyme, which breaks down the disaccharides into glucose, and by inhibiting this enzyme, you inhibit the uh, production of glucose in the intestine, and so the glucose levels in the blood are reduced. A carbose, I can give you good example of that. And you could even inhibit amylases that are secreted by pancreas. So you could inhibit alpha amylase and alpha glucosidase could be your target. So if you have NCEs or phytoconstituents which inhibit these two enzymes, you could always say that these could have good potential as an anti-diabetic agent for that matter. So that's the reason I included these two here. And the information about these. So dietary polysaccharides are converted to oligosaccharides and disaccharides, where alpha glucosidases work on disaccharides and convert it into glucose. So you could inhibit both these. This could be an ideal target to screen test drugs which could have anti-diabetic potential. Could have. You don't know. You can explore anything. So simple experiment. You have a substrate solution. Now substrate is for amylase is always starch. And sample solution. Sample is the one which is inhibiting which is supposed to be inhibiting the enzyme that is converting this substrate into product. So substrate plus test, of course test, you also use a positive control, a standard inhibitor of alpha amylase and a standard inhibitor of alpha glucosidase. you could use that, and you have to use a standard positive control, so that just to ensure that your study is going all right, the design is perfect. Pre-warm at 37, and then you add the enzyme solution, alpha glucosidase or alpha amylase. Either you can buy that, or you can ask for gift samples from enzyme company. So that's the best way to go about it. Mix and incubate at 37 degrees centigrade. Add purified water to make up the volume. Stop the meaning in this, if it is when you put it at 30 minutes at 37 degrees, the enzyme will try to convert it into disaccharides and monosaccharides, and the test substance, if it inhibits the enzyme, will not produce the product. So at the end of 30 minutes, you dilute the whole thing and you put it in boiling water to stop the reaction. And then just read the absorbance. This method is also not new. It is already there in the literature. So it's easy. And from this, you could find out the inhibition concentration 50 of the test product. So for amylase, you use starch, and this is a DNS coloring agent, and for both of them, because this DNS coloring agent, it reacts with the reducing sugars. So in fact, you're measuring glucose here. Measurement of glucose is happening with this DNS reagent. And of course, in alpha glucosidase, you have maltose and sucrose as substrates, disaccharides. And here you have starch as a substrate for amylase. End product for both is glucose. Coloring agent for both is DNS coloring agent. Okay. So you will get some values of alpha amylase and alpha glucosidase inhibition. So IC50 meaning lesser the IC50, powerful is the compound in inhibiting that enzyme for that matter. I'll check if I have some values for you. I may have. Let me check. Now, so we checked antioxidants, we checked advanced glycation end products, we checked in enzyme inhibition assays, we checked two enzymes, amylase and this one, what you say, alpha glucosidase. Now we will check one signaling pathway and one enzyme which is involved in that signaling pathway. So normally what happens, when you have normal sugar level, glucose gets converted to fructose only 3%. So only 3% of glucose gets converted to sorbitol and fructose during normal glycemia, normal sugar level. But when there is hyperglycemia, 30% of the glucose gets converted into sorbitol and fructose. Now, how is this? It is with the help of an enzyme called as aldose reductase. And sorbitol is getting converted into fructose by sorbitol dehydrogenase. So, basically, important is this stage where glucose is getting converted to sorbitol. So, you have to think of aldose reductase as a target. So if you could inhibit aldose reductase, you won't have sorbitol or fructose form, right? And why these are dangerous? Because sorbitol and fructose, both of them are producing osmotic stress, oxidative stress, reductive stress, and glycative stress. Meaning different kinds of oxidative stresses are being produced with this glucose to sorbitol or fructose conversion. So this enzyme happens to be one of the important target for that matter. So inhibition of aldose reductase will also serve as a target for reducing complications associated with diabetes. Okay, you are not looking at glucose level at all. You are simply looking at stopping the complications for that matter. Again, very, very simple. Either you could buy the enzyme or you could homogenate lenses. Remember, and you could get the lenses from the abattoir also. Now, I have written rat lenses, but trust me, you can even use goat lenses or you can use cattle lenses and abattoir if at all you have, or any shop which sells meat. You could get it from there, the lenses, and you could just homogenate them and get a crude enzyme. Procedures are also there to separate aldose reductase enzyme from lenses. So this is a reference, but you could check it. All you do is you get a, I've just given a brief procedure, but you obtain enzyme in this particular manner. So you have the enzyme, the substrate you're going to use is xylose, and the test inhibitor I'm going to use that ESA, EASA. So ethyl acetate extract of Seraca indica, and quercetin I'm going to use it as a standard. And all what you do is procedures are already there. You should just check the decrease in absorbance. It's an enzyme kinetics at 340 nanometers for 3 minutes, and you check the percent inhibition of aldose reductase and calculate the IC50 just the way you would calculate for alpha amylase or alpha glucosidase. So now I have this interesting data. I forgot to put in the uh, alpha amylase and alpha glucosidase. Anyway, if I have, I'll pass it on to Dr. Pawan. So now aldose reductase inhibitory activity, see this is methanolic, this is butanolic, this is pet ether, this is water, and this is quercetin. So if you see that the EASI, ethyl acetate extract, it produced 93% inhibition, which is equivalent to quercetin, which is 94%, literally equivalent. But if you see butanolic and methanolic anyway is better, but meaning comparable. But if you see IC50 of methanolic, it's much higher. But if you see EC, ASI and quercetin, they are both are almost similar. Okay, so this is how you can even grade compounds having aldose reductase activity based on their IC50 values. Okay, now coming to the last aspect of it, it should have come first actually, but then yeah, I thought let me just even take you through this. Now, see, instead of doing alpha glucosidase or alpha amylase or aldose reductase, you have docking softwares available now, and these docking softwares are even freely available. And in that, you have this protein data bank. We call it as PDB. And you have this PDB, and all you need to do is whatever compounds that you want to check for its biological activity, you could just dock them onto these enzymes of interest. And the compounds that show maximum activity, only then you could take it ahead with your even in vitro assays. And what are you going to know from this? You're going to understand from this that XYZ compound has this much of IC50, which is at this particular level as that of standard, and hence it could have or it could not have an anti-diabetic potential for that matter. So don't you think this kind of assays also are equally important? Meaning, as a pharmacologist, I I tell you that none of these enzymes which are responsible for a particular kind of pathological activity, you could choose them. They could be your enzymes of interest. I have also docked NADPH oxidase, and I have even worked wet lab experiments with this. And this NADPH oxidase is one of the important enzymes that produces inflammation. So if you could inhibit that, you could have anti-inflammatory activity in the body. And anti-inflammatory activity meaning reduction of diabetic complications for that matter. As a matter of fact, I have also docked GABA because I've been working on epilepsy, and you could even have few more ligands like alpha synuclein and step 61 for that matter. You could have many proteins are there, so many of them. But just think, what is that protein important in? What is its action? Whether it's a receptor or a ligand or a enzyme, was what is it? And then you could just perform your docking experiment, and then you could give a basic idea to your group that yes, it could have this particular activity for that matter. So, thank you very much for patient listening. This is my current team, and Niyamat has made this particular presentation. So, thank you so much. And yes, we can have question and answers now. I'll just open my this thing also video. One second. Thank God, I was upset because I thought now my throat is really going to give me an issue.
Okay. It was really a nice session, ma'am. So there is a overwhelming response of participant in live chat box. This reflect the actual curiosity and interest of participants. So I have
Please, I'm ready to answer most of them.
some questions here. So
Mhm.
okay, ma'am. With due permission of yours, shall we proceed for
Mhm.
Q&A session?
Sure, sure, sure.
So first question is, how can we correlate neurological disorder with metabolic kind of diseases during research?
Yeah, yeah. See what happens, just think of, uh, as I spoke about AGE, advanced glycation end products. So when advanced glycation end products are there, there is a receptor that the macrophages express, and that receptor is actually called as RAGE, receptor for age. So literally, it is a rage kind of reaction. Total inflammation happens even in the brain, and that's how metabolic disorders can correlate with the neurological disorders for that matter. Because most of the neurological disorders are degenerative in nature, and it is the inflammation that leads to degeneration of the cell. So
Okay.
rage is the
I have another questions. Like, question is, most of the plant and herbs are evaluated and screened for therapeutically active elements. So, how can we decide phytoconstituent for drug repurposing for neuropathy? Like, is there any
for
criteria for it?
neuropathy. Okay. So first of all, you identify what you want to study. If you want to study Parkinson's or Alzheimer's or Huntington's or whatever. And so once you identify that, you should know the full pathophysiology. The thing that I told you right in the beginning, literature survey is most important. So do a thorough literature survey and have a good pathophysiological basis for identifying what you want to study. So once you have pathophysiology, you know what biomarkers you are going to study. So once you
Okay.
know the biomarkers, you can then, whether your phytoconstituents are antioxidants, whether they are inhibiting any particular enzyme.
Yes.
Neurologically also there are so many enzymes, cholinesterase enzyme is there. Anticholinesterases are also used as major therapeutics for Alzheimer's disease. You know that.
Another question is from Sabre sir.
Hmm.
He is my colleague. So, how to find out new bioactive molecule during research, because it is costly to purchase.
Ha. Okay, okay. So you are you want to focus on herbal substances basically, right?
Yeah.
So do you have a nice pharmacognosy department to back you up?
Yeah.
Huh?
Yes.
So
Yes. Yes.
they would be isolating, purifying many compounds. So you have a tie-up with them. So that's how we go about in ICT. So pharmacognosy department, see, their job is not to determine biological activity. The chemistry people should just synthesize the drug, It is the responsibility of the pharmacology to evaluate them. So that's how you could have a correlation, collaboration with them, and you could go ahead and explore the way I told you.
Okay.
Simple thing is go on doing antioxidant activity first. And I have just explained you two antioxidant assays, but there are many more. Ferric ion scavenging activities there, ABTS is there. There are many more. So nitric oxide scavenging is there. So you have just go and have a look at antioxidant activities and what protocols are there, and at MPharm level, meaning as a professor to your students, you just give two molecules and explore them in a particular manner so that you come to one stage, and then you could give it to maybe your PhD students for exploring it for further activities, meaning in vivo. I think at MPharm level, you should not give the students to work on animals, but then it's an injustice to them. So you take a call. Whether you can afford it, then it's good. You can do that. But it's really expensive. And what happens, nowadays, MPharm students are there only for 2 semesters with us. 2 semesters they give exams, and only 2 semesters they are there for research. So in those 2 semesters, we have to train them to handle the animals and then actual experimentation. So instead, I feel these in vitro assays, they do wonder. And I've just
Yes.
explained you one diabetes. You can have as many diseases, as many enzymes, as many targets. And in silico also, you don't give it up.
Yes.
You try thinking of that also.
Okay.
And free softwares are also available. You can
Yes.
do that.
Yes. Okay, it was a really nice session, ma'am. Ma'am talked about 6 steps to achieve your goals in any kind of scientific research. That is,
Don't stop. Publication is must. Remember, patent बाद में.
yes, yes,
At
yes.
least tell the world that this is what I have done.
And ma'am also discuss about branches of experimental pharmacology and important approaches in research, that is in silico and in vitro. Seriously, these are very important research approach nowadays. Ma'am also share information about software availability of toxicological studies, and that will be really convenient to our master student to do toxicity studies, and also talked about docking software and their importance, and share info about several antioxidant assay and their important endogenously present antioxidants. And thank you so much, ma'am. It was a really informative session for students and also for research scholar and also for me, because I work in same area.
Nice,
Now
nice.
now I hand over the session to Anjali Tazampure ma'am for the next and last session of the day. Over to you,
Okay.
ma'am. Over to you, ma'am.
Good afternoon. So let's sail out our program to the third session, the one of the most awaited session of Dr. Kalidas Gole sir. I'll just give a brief introduction about him. Sir has obtained BVSc and AH from Nagpur Veterinarian College, Nagpur in 1989 and MBSc in 1992. His research interests are developing animal models, characterization of spontaneous mutant animals, health monitoring, cryopreservation of germ plasma. He has established and characterized spontaneous mutant mouse model for congenital cataract and microphthalmia condition. And this work was presented in his PhD part, which he received in 2009 via Pune University. He has published 21 research articles in national as well as international journals. He has delivered about 40 lectures in several pharmacy, veterinarian, medical colleges, etc. He is reviewer for more than 5 national and international journals, and he is faculty for the certificate course on laboratory animal sciences, FELASA accredited course, which is first kind of course which is outside Europe and it is conducted in Madras and Madras Veterinary College, Chennai. Every year, this certificate course is conducted. So it is all happening under his guidance. He is serving as a CPC SEA nominee on several IAECs of research institutes. He is ad hoc consultant for Association for Assessment and Accreditation of Laboratory Animal Care International, USA for Asia Pacific region. In 2018, he was awarded with the renowned fellowship by MDI Biological Laboratory, Maine, USA, for attending training course on health and colony management of zebra. What an eminent personality we have with us today for this session. Welcome you, sir.
Thank you, Ms. Tajanpure, for brief introduction.
Yes.
Let me share my first slide, please. Is it visible?
Yes, sir.
Okay. So I express my sincere thanks to Professor Sirsagar and Dr. Udavan for the invitation and giving an opportunity to share my experience in the field of zebrafish, laboratory zebrafish. So I'm also thankful to earlier two speakers for their excellent talks and very useful and informative talks. So Dr. Mali talked about 3 Rs, which is very essential, mandatory in animal experimentation. Tissue culture is actually, when this tissue culture was developed maybe 3 or 4 decades ago, it was thought in a research community that the tissue culture will be substitute for using the animals in research. But each technology has their own limitations, and there is no option other than to use animals in experiments. Dr. Sadhna madam also talk about reducing the animals in experiments, and she insisted to go for the in vitro studies before going actual for the animal studies. But as I told you, yes, at some point we have to use animal models for research. Again, see, all animal models are not ideal for all type of research. So each animal has very peculiar biological characteristics, and that make them suitable animal model for certain human conditions. So I will tell you an example of bulb sea mice. Am I audible?
Yes, sir. So there is just a small request. Will you please
Okay.
switch on the presentation to the slideshow mode?
It is on presentation mode.
Slideshow, sir.
Yeah, it is on slideshow.
Okay, fine, sir. So you are audible.
I will try again because in my screen it is in presentation mode.
Okay.
Let me try once. Now it's okay. Is it in slideshow?
We are not getting it in the slideshow mode, sir. But it's fine, sir. We can read the part on the slide. It's okay, sir.
Okay. So for example, each animal is not suitable for all type of conditions, as I told you. I will give you an example of bulb sea mice. So bulb sea mice is ideal for developing the hybridomas. Because in bulb sea mice, these plasmacytomas are easily developed. No other mouse strain is actually suitable for this work. The other strain is C57BL6 mice. So it is very ideal for diet-induced obesity. So in 2 to 4 weeks, actually, you can induce this model for obesity by giving the high fat diet. So okay, so this is all about the earlier two talks. Switching over to my talk on zebrafish. So I take this opportunity to share my knowledge and experience in the field of laboratory animal science, which I have gained during last 10 years. Actually, I am trained in rodent animal facility, but anyhow, now I'm looking after both rodent facility as well as the zebrafish facility. So I will be talking about a small fish that is a cyprinid fish, zebrafish. The scientific name is Danio rerio, and this is the youngest animal model in the area of biomedical science. So in Western and European laboratories, it has been in use for the last 3 to 4 decades. But in Indian scenario, this model has found place in the laboratories for last 8 to 10 years. Our facilities also developed in 2010, and in many Indian national laboratories, they have established their zebrafish facilities because of very useful features this species has gained naturally. So I will be talking about, it is not a very in-depth talk, so I will be talking about the introductory part of the zebrafish and the useful characteristics of this model for the biomedical research or testing or training or in pharma research. And then housing conditions and genetic manipulations used to make them or to model them for several human conditions and their research applications. So we all are aware about these non-vertebrate animal models. Drosophila melanogaster, you all be aware of. This is very old model, and it is also called as a queen of genetics. So many genetic pathways have been evolved using this animal model. The next one is C. elegans. This is again very wonderful and useful animal models, and still many researchers are using this model along with the other animal models. So C. elegans is mostly used for neurotoxicology, genetic toxicology, and experimental toxicology. And many high throughput studies with the C. elegans are used for targeting the targeting molecular markers that can be used as indicators for the toxicity studies. The third one is earthworm. Again, it is used in many zoology laboratories, zoology colleges. Hello.
Excuse me, sir. Could you please change the slide because the presentation is on the first slide.
It is not moving.
No, it's not moving, sir.
No, but it's moving here now.
No, that is the problem, I guess.
Now.
No, still not moving.
But here it is moving. On my computer, it is moving.
Here it is not even on the slideshow mode.
So let me stop sharing and again share.
Okay, sir.
I don't know. Now is it okay? Is it visible?
It is visible, but still it is not on the slideshow mode. It's not on the full screen.
I don't know because it is changing here and it is on slideshow mode. I don't know why it is not.
Udan sir, can we help him? We will do the next slide for him.
For that, sir will have to share the presentation.
So I will have to email you.
Yes, sir. Yes.
Let me try once again.
Okay. Okay, sir.
Now see that whether it is visible.
Yes, it is visible, sir. Yes, could you please change slide, sir? First of all, could you
Moving?
please next slide? No, not moving. It is on the very first slide.
No, but it's moving here on my PC.
No,
I don't.
not on the YouTube screen. It is not moving, sir.
Okay, so what I will do, I will email you this presentation. Maybe you can share
Yes,
the
sir.
audio.
Yes, surely.
Sir, have you sent that mail?
Yes, yes, just I'm sharing. I'm sharing both my webmail and Google also. One minute.
Yes, sir. Yes, sir.
Just check your email. I have just shared with you.
Yes, sir. We will check.
Have you received it?
Not yet, sir.
It is already emailed.
Yes, sir. We are checking. Sir, have you sent it to Pawan Udan, sir?
Yes.
Okay, sir. Okay, sir. But not yet received, sir.
I don't know. I have sent it through my TIFR email. Now I am sending through Gmail. So
Okay, sir. Please, sir.
yeah, I have sent using Gmail account. Just check now.
Okay, sir. Okay. Yes, sir. It is received.
Okay, now you can.
Okay, sir. Within a second, we will share it.
Okay, sir. Now we can go ahead. We can
Okay,
see
sir.
the screen as well. Sir, change the. Could you please change the slide? Yes.
Yeah, second slide. Okay. Yeah. So I was about to tell about earthworm. So earthworm. Uh, Hello. So earthworm is also being used in many research laboratories. In Chinese medicine, it is considered as a biomolecule. And as per the Chinese medicine, it has been used for treating around 19 diseases. So it is also very useful. Next slide, please. So we should be grateful to these vertebrate animal models. These are the wonderful creatures of nature, ranging from tiny mouse to large animals such as the horses. These animals have significantly contributed in the areas of fundamental research, training and testing, and many breakthrough could have been possible in discovering many pathways, drugs and vaccines, biologicals. So it's been now more than 100 years. Mouse has been in use in research, especially for genetics, cancer biology, and um, cell biology. The other model is rat. Rat is bigger than mouse, and it's very useful for neurobehavioral studies, toxicity studies. Guinea pig is very susceptible to vitamin C, and it's being used for scurvy, as well as it is also susceptible to tuberculosis, and for developing the tuberculosis, guinea pig is used. For complement research, it is again a very good model. Rabbit is used in pharma industry for pyrogen testing. It is also ideal model for atherosclerosis research and production of the polyclonal antibodies. Cat is again used for behavioral studies and for several assays of hormones. Dog is used for cardiovascular studies, especially for organ transplant, and also it is used for behavioral studies and toxicity studies. Monkey is again very close to human being, and very many neurodegenerative diseases, Alzheimer's, Parkinson's are being studied in monkeys, and malaria is another disease which is studied in monkeys. Sheep is again a very useful model for cardiovascular studies, especially for angiography studies and stent or heart transplantation. There are transgenic sheeps have been developed which secrete the enzymes in their milk, and in natural condition we get these enzymes in very trace quantity, but using the transgenic sheeps we can actually get these enzymes in liters. Horse is again, it is used for sera production, biological production, and venom, and is a tetanus toxicity studies. So next slide, please. So fishes are worthy of experimental and observational research. They are very useful indicators of environmental quality and ecological integrity, and their individual adaptation and physiological specialization make them useful models for physiological and biomedical models. So different fish species are being used in biomedical research. Among them are zebrafish and medka fish. These two species are very popular. And zebrafish is Indian river fish, whereas the medka fish is originated from the Japan. Other fish are guppy, goldfish, then carp, common carp, rainbow trout, pufferfish, catfish, and minnows. But these are not widely used as compared to the zebrafish and medka fish. So today I will be talking about the zebrafish. Next slide, please. So if you trace the origin of zebrafish, it is found in the Ganges and Brahmaputra river. It is also found in some parts of Pakistan, Nepal, Bangladesh, and Myanmar. The natural habitat is tributaries of Ganges and Brahmaputra. It is also found in slow streams of water and paddy crops and lakes. For the first time in 1822, it was reported by a British army personnel based in West Bengal, Dr. Hamilton. Then in 1970, Dr. George Stressinger at the University of Oregon, USA, for the first time he used this model for making different clones and for the genetic analysis. Recently in 2013, Professor Muthuswamy from Kerala University, he surveyed many parts of India and he found total 21 different wild type populations. Next slide, please. So zebrafish has been in use since 1950, but the tremendous growth in the publications using zebrafish model has been seen after 1990. This is because in early 90s, two major zebrafish mutant screens were conducted, one at Max Planck Institute Tübingen in Germany, and other is Boston Children's Hospital, Boston, USA. And the product of these two major screens are around 15,500 mutations found in around around 400 genes, and these genes are actually involved in mechanisms such as the development and organogenesis. Next slide, please. Okay, so zebrafish share more than 70% homology with the human. Next slide. Okay, so it share more than 70% homology with humans. Being vertebrate animals, it is very close to human being as compared to non-vertebrate animals such as the C. elegans and Drosophila. And many genes, genomics, and pathways are actually conserved between the human and zebrafish. Being very small animals, it is easy to breed in the laboratory conditions, and large number of the zebrafish can be kept in a small space. Compared to the rodent facility, maintaining the zebrafish is less expensive. Next. It is very easy for the genetic manipulation in zebrafish, so that many mutant models can be developed using transgenesis or knockout technology or chemical screens, and a large number of the mutants can be generated. Females are very fertile. They produce large number of embryos. It can range from 50 to 400, and more than 80% of these embryos are fertile. So the advantage is that large number of the embryos are available, especially for the high throughput studies. the fertilization and development takes place outside of the body. So it is very easy for the researchers to access this model, especially in the developmental studies or embryology studies. If you use the mammalian model, you have to sacrifice the mother, and then it attracts the ethical issues. Whereas in zebrafish, actually, we have no that issue. Means without ethical issues, you can use these embryos for such type of studies. And these embryos are crystal clear, transparent during the full development stages. You can say the early development up to 5 days. These are very clear. So you can easily visualize the development of tissues and organs with a simple stereo microscope, as well as you can actually see the effects of drug on the developing organs or tissues. You can also see the toxicity of chemicals on the tissues and organs. Next slide, please. First one. Okay. So coming to housing fish in the laboratory condition. So good quality water is the major infrastructural requirement for any aquaculture facility, or you can say the zebrafish facility. The major challenge in fish housing system is maintaining the quality of water constantly clean. Because if the quality of water is not clean, then these fish are actually suffer from the toxicity of their main metabolic products, such as the nitrogenous products. So fish are actually reared in non-circulation system as well as the circulation system. So non-circulation system is a conventional method of rearing the fish. You can maintain the fish in simple glass aquarium, or you can also use mice and rat cages. And fish are raised here in static water. So there is no recirculation of the water, and no biological filters are used. Here you have to do the manual cleaning and water exchange. This is a low cost setup to establish and maintain, but the quality of the water cannot be guaranteed, and the stocking density you cannot actually increase more than 1 or 2 fish per liter of water. Next slide. The recirculation water system is the modern age housing system for zebrafish. Here the water is continuously recirculated 365 days after passing through a series of filters and then the sterilization. So many mechanisms are inbuilt, like automatic water exchange, then water aeration, degassing, and then the filtration system, sterilization, heating and cooling, and then the light and sound alarm system. So these features actually inbuilt in system, and it actually reduce the manpower because many functions are done by system itself. And here the most important thing is it's an absolute used for prevention of the contamination. So there is absolute no cross contamination between two neighboring tanks, because the water they get from the system is individual, as well as the overflow water is actually poured separately. So absolute there is no mixing of the water. And as I told you, since it is a machine operated system, fully automatic, there is a reduction in the manpower. Manpower you need just to feed the fish, and just some other you can see some checking the sick fish, and just do some buffers, making the buffers. But other major infiltration filtration and sterilization, other major work is carried out by the system. So the upper one is a single standalone unit, and below is the multi-leaking system. So as per your requirement, either you can get one single unit, or you can have a big facility where multiple standalone systems are interlinked, and they get the filter water from a single filtration assembly. Next slide, please. So characteristics of the zebrafish reproduction, if your fish are healthy and your feeding regimen is good, then sexual maturity takes place after 60 days. However, you can use these fish after 3 to 4 months for mating. So there is a simple difference between the male and female. Males are slender and they have orange or you can say the golden color stings on their strips, whereas females are like having a enlarged abdomen, and their strips are you can see white strips on the abdomen as well as on the fins. So this is simple difference you can use for actually identifying the male and females. Breeding is decline after 2 years. So the major potential breeding period is after 4 months till you can say 1.5 years, or at the most 2 years. After 2 years, they develop many diseases. So it is better to remove these fish after 2 years. Lifespan is till 4 to 5 years, but those very useful lines like transgenic or knockouts, if you really need, you can maintain till 4 to 5 years. Otherwise, white type stock you can clean after 2 years. Length is 3 to 4 centimeter and weight is around 0.4 to 0.6 gram. As I told you, they lay in nature in stagnant water bodies, ponds, rivers, paddy, and also this fertilization and development takes place in the same habitat. The female is very fertile. As I told you, several eggs are produced per female, and more than 80% eggs are fertile. In wild type population, sex is determined by the chromosomes. So there are sex chromosomes for determine the sex. Whereas in laboratory setup, now this fish has been used for last 40 years. So it is noticed that they have actually lost this locus, and now the sex is determined by the environmental factors like low oxygen density, stocking density, high cortisol, and stress hormones. So this is very surprising thing in zebrafish. Next, please. So zebrafish grow and develop through 4 different stages. You must be knowing these 4 stages because especially for planning your experiments on and ethical issues, these stages are important. So first stage is embryo stage, which lasts for 1 to 3 days, and during these 3 days, zygote formation takes place. Then cleavage, blastula, gastrula, segmentation, and hatching. So hatching takes place between 48 to 72 hours. The next stage is larva stage, which starts from 4 days to 29 days. So again, it is subdivided into swimming larvae, which is for 4 to 6 day post fertilization. You can say a swimming larva, and then 7 to 13 day post fertilization, you can say early larva, and 14 to 29 day post fertilization mid larvae. The third stage is juvenile, that is for 30 to 59 days, and after 60 days, you can say it is adult. In some laboratories, they call adult after 90 days. So here, for ethical issues, now CPCSA has also come with the guidelines for fishes. It is recently published, maybe in March or April 2021, they have declared these guidelines for the fishes. Sorry. So till that time, we were following the NIS guidelines or PHS policy or OLA guidelines. So as per OLA guidelines, embryos for first to 3 days, this stage is not actually living animal. So these using embryos in the research are actually exempted for the counting purpose. So you cannot count it as animals. You have to approve the project, but these embryos cannot be counted as animals because they are not considered as a living animal. So living animal is considered only after hatching, when they come out of the chorionic membrane. So then 29 days. So they are saying after fifth days, because cognizance is developed on fifth day. So then after fifth day, you can actually count them as animals, and you can maintain the records for a number of fishes or larvae are used in the research. That is as per the NIS guidelines. In our guidelines, I have seen they have not mentioned this. So maybe someone will have to talk with the CPCSA again for this clearance. Okay, next slide, please. Coming to the experimental approach for genetic manipulations. So various methods have been established for the genetic manipulation of zebrafish for producing various mutants. So chemical mutagenesis using the ethyl nitrose urea is most popular. This was used in early 90s when I already actually mentioned two big mutagenesis screen in early 90s. So this this chemical was used for mutagenesis screen. It is used for the random mutagenesis, and then the progeny in F3 generation are actually analyzed for the genetic abnormalities. The second one is the production of haploid and diploid embryo. So this is very useful feature in zebrafish. You can produce the haploid embryos, you can make them the diploid using a simple technique. The third one is a transcription activator like effector nuclease and the zinc finger nuclease ZFINS. And then again, most popular technique is the knocking down of the genes using the morpholino. This is also most popular. It is a reversible effect actually. It inhibits the protein synthesis. And the most powerful technique recently developed for which in 2020 this Nobel Prize is awarded, that is the CRISPR. So it is most powerful technique for genome editing. So next slide, please. So chemical mutagenesis using the EMS and ANU are used for the random mutagenesis in zebrafish. This approach is also used for the forward genetic studies. The technique is simple. Males are actually exposed to ANU, and then those males are crossed with the wild type females. And then the embryos are produced using this cross, and they are raised till the sexual maturity. And once they attain the sexual maturity, again they are crossed with the wild type animals. So in F2 progeny, you get 50% wild type population and 50% heterozygous population. So now using this F2 sibling, you can make the cross to produce the F3 generations. So in F3 generations, if both the parents, male and females, are heterozygous, then you can anticipate 25% wild type population, 50% heterozygous population, and 25% mutant population. So this will also confirm that both parents are heterozygous. If you get only 50% wild type population and 50% heterozygous population, it means both the parents are not heterozygous. So those 25% of the mutant phenotypes are used for genetic analysis. So what are the genes those are mutated, and what type of conditions they are actually showing, or what type of phenotypes they are showing after this genetic mutations, and whether those conditions are mimic the human conditions. If they are actually similar with the human conditions, so those can be used as a model for studying the human diseases. Next slide, please. So as I told you, ploidy mutations in zebrafish is very popular, or you can say it is easily done. So in vitro production of haploid and diploid embryos is possible in zebrafish due to its external fertilization. It is a powerful tool for mutational analysis as it enables the identification of recessive mutant alleles present in the F1 generation. F1 generation female carriers following mutagenesis in the parental P generations. So this approach eliminates the necessity to raise the multiple generation F2 like F2 and F3, which involves breeding of the mutant families, thus saving the researchers time along with reducing the needs for zebrafish colony space, labor, and husbandry cost. So zebrafish embryos can survive up to 5 days in the haploid state. And the phenotypes are like phenotypes you can see in haploid condition is body is very short, it's very thick, and it's a pericardial edema. But this haploid state will not continue, and then those embryos will die after 5 days. Next slide. So this figure shows how this haploid and diploid screen is done, and how in haploid screen we can reduce the continuation of the you can eliminate the continuation of F2 and F3 generation. So in left side, I'm sorry, I can't use my mouse. So in left side, this is a typical diploid screen, which I have already explained in the first slide, where you have to raise the F1 and then F2 and then F3 generation. Whereas in haploid screen, after the parental generation, you take the females, obtain the eggs, and then these eggs are fertilized using the in vitro fertilization with the sperm, which are UV irradiated. So when you actually irradiated the sperms, it kills the genetic material of the sperm. However, their fertility potential is intact. So they will fertilize the embryo, but only the genetic material from the female will be inherited to the next generation. So in F2 embryo clutches, you just see the 50% mutant. So it means if you get the 50% embryo clutches, if you get the 50% embryo, sorry, if you get the 50% embryo clutches with the mutant, it means that female is carrying the recessive mutations. And that's how that female can be then used for recessive mutation analysis. Next slide, please. Using morpholino injections for the reverse genetic approach is again most popular, as I told you. So it is a simple injections of morpholino in one cell embryos. So morpholinos are nothing but it's a neutral non-ribose backbone displaying 4 DNA bases, stably base pair with RNA, but are resistant to degradation, resulting in the penetrant gene that produce knockdown effects. It is not knockout effects, it's a knockdown effect, it's a reversible effect. And morpholinos function by forming the heteroduplexes with the target transcript to interfere the protein synthesis or splices. So morpholinos demonstrated by targeting the translation site start for a number of developmental genes, where treated embryos mimic the phenotypes observed in correspondence with the corresponding mutant alleles. So this is simple. So during the early embryonic stages, you can see the effect of this protein synthesis on the phenotype, and then after 5 days, you can that effect is reduced. So for early embryonic development, for producing the different phenotypes as per your experimental need, you can design your morpholinos. Next slide, please. And this is the most powerful technique, as I told you, for genome editing, CRISPR. So here, actually, you can delete a single nucleotide, or you can remove a part of your gene sequence. You can add the gene, or you can repair the gene. So several mutant in almost all species of the animals have been produced using these techniques, and in future, so this technique could be used for repairing the genetic disorders in human. So that's the potential of this technique. Next slide, please. This is not genetic manipulation, but this is a cell transplantation technique. Again, it is well established in zebrafish. So cancer cells can be transplanted in adult stage as well as in the embryonic stage. So in the adult transplantation, you can actually remove the neoplastic cells from the adult, and then it can transplant into the another adult which is irradiated to accept the graft, and this neoplastic or cancer cells can actually grafted in the adult, and it can also metastasis. The other approach, actually, the human cancer cells with the fluorescent markers can be co-culture with the developing embryos, 3 to 4 days zebrafish embryos, and then these embryos will take these cancer cells and it will grow. So these are the two approaches for actually developing the zebrafish cancer model. Next slide, please. Last part of my talk, the research applications of zebrafish model in various areas of the biomedical research or toxicity testing or drug discovery. So next slide. So zebrafish has been a traditional model for studying the vertebrate embryonic development because of this external fertilization and external development. Many processes that takes place during the early development, like tissue morphogenesis, organogenesis, or apoptosis, or cellular and molecular mechanisms and signaling pathways can be studied using this model and models, and it's easy access easy access to the embryos without any ethical issues. And the data obtained from these early embryonic studies can be compared with the other vertebrate animal models, and then you can also do the comparative analysis. The one more area, that is the maternal genes. So maternal genes which are involved in the early event of life can be actually studied, because see, when the fertilization takes place, before the fertilization, eggs, how the egg is prepared for the fertilization, and also how it helps or it support the early embryonic development. So what are those maternal genes which are actually responsible for these early events of life are actually being studied using zebrafish embryo. Next slide, please. In the area of drug development or drug screening, zebrafish is again playing a crucial role. So with the advancement in rapid synthesis techniques, large numbers and diverse compounds are synthesized, and using these compounds in high throughput experiments, large preliminary hits are actually generated. So to validate these preliminary hits, you need a model. Normally, mammalian models are used, but they are very slow and very costly. So now zebrafish assays are being used for validating these early hits, because these assays can be done in 96 well plates. So it is very easy for dosing and less drug is required. The most important thing is you can actually visualize the effect of these drugs on the developing tissues or developing organs. So this is most important, which is not possible in other animal models. And the in vivo zebrafish assays for drug effect on the anagenesis and apoptosis can also seen, also can visualize using this model in 96 well plates. Next slide. Now it's been more than 20 years. Zebrafish model has been used in chemical toxicity, determine the LC50 and also teratogenicity. So this is because so many advantages of the zebrafish model, like availability of large number of the fertilized embryos from single female, and the zebrafish bioassays are very cheaper and faster than the mouse model. Embryo assays are carried out in 24 and 96 well plates, easy to dose and less dose is required. And visual assessment of the toxic effects of compounds on the developing organs, tissues can be easily seen using a simple stereo microscope. And the four parameters of lethality are well demonstrated in the zebrafish, like embryo survival, behavior, and organ malformation in zebrafish. And zebrafish embryos have been shown to be useful model for detection of the direct acting teratogens. So next. So with these useful features, OECD has already validated zebrafish model for such types of studies, and there are number of tests like 203, 204, 210, 212, 215, 229 used for the various toxicity studies. And in the right column, the stages of the zebrafish which are actually suitable for carrying out such toxicity studies. Next slide, please. So this is fish embryo toxicity 238 to determine LC50. Newly fertilized embryo zebrafish eggs are exposed to the test chemical for a period of 96 hours. 5 concentrations are used and 4 apical observations are recorded as indicators of the lethality after every 24 hours. At the end of the exposure period, acute toxicity is determined based on a positive outcome of any of the 4 apical observations recorded, and the SC50 is calculated. So on the right side, you can see there are 4 indicators of the lethality, like coagulase of the fertilized embryos, or you can also say death of the embryo, and then loss of somatic formation, then the loss of detachment of the tail bud from the yolk sac, and the lack of the heartbeat. So these are the major 4 observations which are seen or monitored during the toxicity studies. And here 20 embryos are used for each concentration, and then you can use 5 concentrations for determining the LC50. Next slide, please. This is another test, test number 203, fish acute toxicity test using adults to determine LC50. So here, adult fish are being used, 7 fish per concentration are used, and then the observations such as the mortality, abnormal appearance, and abnormal behavior are monitored. And those fish which are survived are euthanized, and then you take the weight and length of the animals, and based on your observation, LC50 is determined. Next slide, please. This is another test. It's a test number 210, toxicity testing using the early embryos. So here again, this test is used for lethal as well as sublethal effect of the chemical agent. So here again, 5 concentrations are used in 4 replicates. 20 embryos are used for concentration, and then 4 replicates. And the observations such as the stage of embryonic development, hatching and survival, abnormal appearance, abnormal behavior, weight and length. So these observations are made during the studies. Again, this is a 96-hour study, and then you can see the toxicity of certain chemicals. Next slide. Next slide, please. This is one paper I have got recently. So zebrafish has been used to test the toxic effect of a biomolecule which is potential for the multiple sclerosis disease. So in Brazil, there is one venomous toadfish. So this biomolecule is present in that toadfish, but that peptide is showing the anti-inflammatory and anti-allergic drug. So they have used zebrafish, and they found that this is a potential model for testing the toxicity of that biomolecule. Yeah, so this is about. So next slide. This is one more paper, and it's very interesting. Dr. Mali has also talked about the 3D printing technology. So this technology is gaining popularity. It is very fast and cost effective and highly customized for fabrication. So the parts we use in 3D printing, we do not know the toxicity of these parts. So in this paper, they have studied or they have tested these parts, 3D printed parts for the toxicity using the zebrafish embryos, and they found these parts are toxic. And they also actually suggested treatment for these parts before disposable. So simple, you can expose these parts to UV light before the disposal. Next slide, please. Zebrafish is also again proven model for behavioral screening assays. So this is actually well studied to discover the genes involved in the proper execution of behavior programs, to identify factors required for neutral cell-fed decisions, differentiation, exome-guided and synapse formations. Mutants obtained in a behavioral screen provide an alternative reasoning techniques complementary to the surgical ablations. So, in many behavioral screening, especially in neuropharmacology, zebrafish has been used. I have seen in a few pharmacy colleges such experiments are being carried out. Next. So, these are behaviors which are also very common in human being are exhibited by the zebrafish larvae, especially the larvae below 8-day post-fertilization are being used for such studies. And then the behavior like swimming and motility, photoentrainment and circadian regulation of the activity, sleep, auditory fast start, touch evoke response, and then optometric response, eye movement, and pursuing the visual motor. So these behaviors are actually studies using the zebrafish model, especially the zebrafish larvae below 8-day post-fertilization. Next slide, please. And this is a typical setup used for video recording or video tracking of this 96 well plate assays, or even adult assays. So here, it's a 96 well plate where the zebrafish embryos or larvae are treated with several compounds simultaneously, and there is a camera, overhead camera. So these behaviors are actually trapped by this camera, and then it is processed in the CPU, and those behaviors are actually display on the monitors. So you can analyze the behavior of different drugs or different molecules on the zebrafish embryos in 96 well plates. When you study the adult, you have to just replace this 96 well plate with the tanks and then the fish. Okay, and these are different stages of zebrafish which are actually used for feeding purposes. The idea of showing this slide is because the stage from 6th day to 60 days, there is a fast growth in the zebrafish larvae. So this stage can be used for testing the different food supplements, hepatitis, or other food or nutrients, you can say. Yeah, next slide. Again, zebrafish as a model for obesity and diabetes. So, can you make it bigger? Actually, I can't. So, zebrafish is an attractive model system for studying the metabolic diseases because of the functional conservation of the lipid metabolism, adipose biology, and pancreas structure, and glucose homeostasis. It is also suited for the identification of the novel targets associated with the risk and treatment of the obesity and diabetes in humans. So many research laboratories now they are exploring this model for obesity and diabetes. Next slide, please. Next slide, please. Yeah. In cancer biology and therapeutics, zebrafish is again used for last maybe 20 years. It responds well to carcinogenic chemicals leading to development of tumors. And cancer in zebrafish can also be induced by transplantation of the mammalian cancer cells as well as transfer of the oncogenes using the transgenes technology. The most surprising thing is that the zebrafish cancers and human cancer, they show the identical histological pictures. And these cancers like hepatocellular carcinoma, seminoma, thyroid neoplasma, intestinal carcinoma, and sarcoma, these are spontaneously develop in zebrafish, as well as it can be induced using all these chemicals or cells, cancer cells, or oncogenes. Next. Next slide. So in 18 years period, yeah, there is a tremendous growth in using the zebrafish for cancer model. So this is a key milestone achieved during the 18 years. So for the first time in 2013, first oncogene-driven zebrafish cancer model is developed. In 2005, first tumor suppression driver transgenic zebrafish. Actually, I am not able to read this slide. It's very interesting. It is in slide show mode. No? Yeah, please. Okay. So in 2005, first tumor suppressor driven transgenic zebrafish cancer model has been developed. During 2006 and 2007, first reports of human cancer cell xenografts zebrafish cancer model is developed using the human xenograft. In 2011, co-clinical trials with the mouse model of cancer and patients with the cancers is done. Whereas in 2013, zebrafish is the first vertebrate engineered by the CRISPR technology. The same year this technology discovered, in the same year this zebrafish has been used. Zebrafish has been developed for the cancer model. Then first reports of PDX, patient-derived xenografts in the zebrafish embryos. It is done during the period of 2015 and 16. And during the period of 2015 and 17, first report of drug testing in zebrafish embryo, PDX, is reported. In 2018, combinatorial rapid genetic modeling using the CRISPR-Cas9 technology has been reported. And in 2019, immunocompromised adult zebrafish developed that can engraft the human cells. And first prospective clinical trial on the predictive power of the human organoids in cancer patient has been reported. So in 18-year period, you can see a tremendous development in using the zebrafish in the cancer research. Next slide, please. So angiogenesis is again very important studies in cancer pathogenesis. So angiogenesis is the formation of new blood vessels from existing ones. It plays crucial role in embryonic development, tissue repair, and also diseases. If you see the therapeutic angiogenesis, the major goal is in the field of regenerative studies. Inhibition angiogenesis is a key therapeutic target to inhibit tumor growth in cancer biology studies. And during the last decades, the understanding of cellular and molecular mechanism involved in this process has been a matter of intense research. In vitro and in vivo models have been established to visualize and study migration of the endothelial progenitor cells, formation of endothelial tubules, and the generation of new vascular networks, while assessing the conditions and treatments that either promote or inhibit such processes. So zebrafish larvae is very potential for using angiogenesis studies because during early embryonic development, especially during the first 4 to 5 days, you can see this vasculature with a simple microscopy and with using a simple dye. So several studies on the angiogenesis and apoptosis in the zebrafish models have been published. Next slide. So this picture shows, you can see this is a trunk vasculature. It is seen after in 72 hours zebrafish larvae. This is the dorsal one is the aorta, and the ventral one is the vena cava. And here you can see the trunk vasculature, which are actually using you can be visualize using the fluorescent or dyes. So many anti-angiogenesis drugs can be tested using this model, as well as you can see the effect of a drug which actually promote the angiogenesis. Next slide, please. This is one more paper recently published. So in our body, cells are moving throughout the life. Cells development till the age wherever there is a requirement of cell, especially in later stages for healing the wound. So how these cells are moving in our body? So research are being done, how these cells are moving, whether there is any mechanical forces which actually push or pull these cells. So now this paper describe the usefulness of zebrafish embryos for studying the mechanical forces in our body, which is used for moving the cells. Next slide, please. In cardiovascular diseases, again, zebrafish is very useful model. Especially in regeneration of the heart, it has tremendous potentials. And cardiac damage can be created by cryo-injury, as well as surgical removal of some part of the heart, especially the ventricle. And these are the conditions like dextrocardia, aortic valve stenosis defect, dilated cardiomyopathy, mitral valve malformation, transposition of great arteries, double outlet right ventricles, hypertrophic cardiomyopathy. So these are common conditions seen in human. And similarly, these conditions are also seen or induced in zebrafish, and then it can be used for studying the human conditions. So, yeah, next slide, please. So this is one paper they have used zebrafish for regeneration studies. So cardiac injury in mammals and amphibians typically leads to scarring. with minimal regeneration of the heart muscle. However, they have demonstrated using a zebrafish model. So what they have done, they have removed 20% of the ventricle using surgery, and then they found in a period of 2 months there is a complete regeneration of this heart and a robust proliferation of the cardiomyocytes. The interesting thing is the ECG of zebrafish and human, they are matching, whereas the ECG of rodents and zebrafish never matches. Next slide, please. Now it is established fact that zebrafish are survive even at low oxygen level. So how it happens actually? What are the type of genes which are expressed during this hypoxic stage in zebrafish? So now zebrafish is again being used for hypoxia research, especially those larvae, because in larvae even they survive at 2 milligram dissolved oxygen per liter of water, where in adult the normal requirement is 6 to 8 milligram or 80 to 100% saturation. So again, this is very interesting area for research where zebrafish can be used. Next slide. In the area of leukemia, again, there are lots of publications. You can go through the Google and see the zebrafish are used in hematopoietic disorders. Next slide. In the area of tuberculosis, this zebrafish can be infected with the natural infection of tuberculosis. The strain is Mycobacterium marinum, which is very close to Mycobacterium tuberculosis. And experimental infection can also be induced in zebrafish. So again, it is a very useful model for tuberculosis research. I have known these two groups. One is Dr. L. Ramakrishnan at the Cambridge University. You will see lots of papers on tuberculosis research using the zebrafish. And other group is at BITS Pilani Hyderabad campus and Dr. Reddy's laboratory. So this paper is published in 2014. Sorry, previous slide. So this paper is published in 2014. They have optimized and validated Mycobacterium marinum induced adult zebrafish model for the evaluation of oral anti-tuberculosis drugs. One more model for zebrafish has been developed at IIT Kanpur, and this paper is published maybe in 2019 or 20. Next slide. So coming to the ethical issues using the zebrafish in research. So recently in March 2021, CPCCI has come with the guidelines for the experimentation on the fishes. You can go through these guidelines. So for the facility registration with the CPCCA, yes and no. When you have a rodent facility or other animal facility which is registered with the CPCCA, in that case there is no need of facility registration. You can add this species as one of the animal models in your research. However, if there is no other facility and you have only zebrafish facility, then yes, you have to register with the CPCCA. IAC approval, yes, you need IAC approval. This is required for actually funding purpose. Because now almost all Indian funding agents, major funding agents like DST, ICMR and DBT, they are asking for the ethical approval, as well as this is required for publication of the research article, because all international journals, they are asking ethical clearance for using the zebrafish in your animal experiments. For exchange between two institutes or for export or import, you have to follow the RCGM guidelines. So these guidelines are again recently come in 2020, and you have to approve your proposal with your IBSC, Institutional Biosafety Committee. and then you can exchange these models as well as any material, biological material related with the zebrafish. Next slide. At the international level, there are OLA guidelines, then the guide for the care and use of laboratory animals, and then the PHELASA guidelines. So you can follow all these guidelines. Like I told you, for counting the number, especially during first 3 days uses of embryos in your experiments. So now the debate is going on at NIH and OLA, whether this period of 5 days can be extended to till 7 days, because there are some reports that cognizance is developed at 7 days, but there are also some reports that cognizance developed at 5-day post fertilizations. So they have also asked your suggestions. Now the debate is going on NIH, and maybe those are interested, I can share that link. So September is the deadline where you can actually suggest your suggestions, and then they will come up with the final guidelines, whether the larvae behind 7 days can be counted as animals, or the larvae behind 5 days post fertilization can be counted as a live animals. Next slide, please. This is regarding the source of zebrafish at international levels. So ZIRC is the main resource center, zebrafish international resource center at Eugene at Oregon University. So you can see this is a Medina or a Kashi of the zebrafish, like Jackson Laboratory for the rodents. You can get everything. You can get everything related to zebrafish. You can get the zebrafish wild type strains. You can get the zebrafish mutant, transgenic, and you can get the antibodies, markers. Even you can get your various protocols for maintaining the zebrafish, or various primers related to the zebrafish research. The other source is the European Zebrafish Research Center, KIT. It is again a similar resource for the zebrafish studies. The third one is the Max Planck Institute for Developmental Biology. It is at Tübingen, Germany. So they also maintain several zebrafish lines, as well as various research material associated with the zebrafish. Next slide. This is our zebrafish facility at TFR. So we have a holding capacity of around 50 to 60,000 adult fish. And maybe this is the second largest facility in India. The first one is at IGIB in New Delhi. And this is a centralized facility. It is fully automatic facility. I have divided this facility in quarantine, and then the nursery and the adult maintenance. Why this is necessary? Because as I told you, these diseases are very common, and those diseases are developed after one year or 2 years. So better these 3 areas should be separated. One is quarantine, like when you get embryos from other site source or adult fish, you have to keep in the quarantine period. Then you get the embryos from those fish, release those embryos, and then introduce into the nursery. So nursery we use only for raising the nursery up to 2 months old. So from 6-day embryos to 2 months old, we raise in the nursery. And in adults, when they cross 2 months, we maintain them in the central facility. And these all facilities are physically separated, and their water supply is totally separated. No, there is no mixing of the water. Next. Coming to the last part, summary is zebrafish, a freshwater cyprinid fish is easy to breed and maintain in laboratory environment. Housing and maintenance is cheaper than the rodent facility. Several biological characteristics have made it popular to involve in various areas of biomedical research. Zebrafish has been occupying a place in drug screening and chemical toxicity studies because of cheaper and faster assays. Zebrafish has proven animal model for human diseases, and with the advent of genome editing techniques, zebrafish can be model for several human disorders. So this is all about my talk. I'm thankful to all audience and faculties and organizer for giving me an opportunity to share my experience. Now we can have a question answer session.
Thank you, sir. What a power-packed session it was. There is massive response coming from the audience since you started the session. We have a lot of questions in the chat box. I'll read some questions for you, sir. Shall I proceed with it?
Yeah, sure.
Okay. So first question is from Rohan Bhadange. He is asking, how can I approach to various laboratories in India for my PhD work? Sir, he might be interested to do PhD in zebrafish model. So where
Okay,
are
so as I told you, the major Indian laboratories, like I told you, TIFR, IGIB, NCBS Bangalore, CCMB Hyderabad, ILS Bhubaneswar, these are the major national laboratories. Even Dr. Reddy's lab. So they have well-established zebrafish facilities, and if you want to do PhD, you have to come through CSR NET set examination. TFR also conduct our own entrance test for the PhD, and then you can you can appear for that entrance test. If you qualify, you can work with in TFR Mumbai campus or NCBS Hyderabad campus or Hyderabad campus. Sorry, NCBS Bangalore campus or Hyderabad campus. So we have 3 campuses in India, and we have well-established zebrafish facilities. Even if you go to IGIB New Delhi, where zebrafish facility is there, and then you can work with the zebrafish.
Okay, so next question is from Dr. Anil Kumar. He is asking to whom to approach for authentication of zebrafish.
Authentication in the sense?
Like whether the strain which is purchased from certain source, so whether it is authenticate genus species or not.
Yeah. So I will tell you. So there are well characterized wild type strains, and then the several. You can see there are more than 4,000 zebrafish lines nowadays available. We have around 50 to 60 different lines of zebrafish. We have only 3 wild types, whereas rest of are all transgenic and knockout, are developed through CRISPR technology. Okay. So for wild types, these are the well-characterized strains we got from the Tübingen Max Planck Institute, like Tübingen, AB, WIK, or you can get from the reliable resource. As I told you, if you want to import, then you can import from the JRC or KIT or Tübingen. So those are actually well-characterized strains. I have seen people in many pharmacy colleges or zoology colleges, they are getting zebrafish from the ornamental fish pet shops, but they are not well characterized. A lot of mutations are there, and these are actually contaminated. So but there are certain studies, you can go with these strains, but if you want to cutting edge research, especially genetic screener, then you should go with the well characterized lines.
Okay, so next question is from Kolkata. It's Yadu Nandan Day, sir. He wants to ask that what are the various important parameters which we should measure during maintenance of zebrafish?
Okay, very good question. Actually, I avoided that part. It's a huge this thing. So it's a major husbandry part actually. So water quality, as I told you, water quality is most important in maintaining the zebrafish. Otherwise, they will actually suffer from their own metabolic product. So what happened when actually you okay? I will go to one of the slide. No, it is with you. No? So anyway, I will tell you. So biological filtration is most important part in zebrafish. Okay. So what happens when you maintain the zebrafish in your tank? So you provide them feed and oxygen. Okay. In return, zebrafish produce ammonia, fecal matter, and carbon dioxide. Okay. And there are some uneaten feed in the tanks. So this uneaten feed and your fecal matter, they generate the ammonia. So again, major part of the ammonia is produced through the gills while respiration, and ammonia is very toxic to zebrafish. So there is biological filtration in the this automatic aquaculture system, like I explained, that standalone and multi-linking system. There is biological filters which harbor two sets of nitrifying bacteria. One is the nitrosomonas bacteria, and other is the nitrobacter. So the first set of bacteria, that is nitrosomonas bacteria, they convert ammonia into nitrite. So nitrite is again very toxic to fish. So second set of bacteria, that is nitrobacteria, they convert nitrite into nitrate, and nitrate is very less toxic to fish. Okay, so if you maintain your zebrafish facility with the 10% of the water exchange over a period of 24 hours, then you can actually reduce this toxicity of the nitrogenous compound. Coming to his question, so you have to see the parameters like temperature. Water temperature is most important, which can range between 27 to 29 degrees centigrade. Then pH. pH is most important. Optimum pH should be around 7.5, but it can range between 6.8 to 8. Okay. If the pH goes above 8, it will kill all the nitrifying bacteria, and then your biological filtration will be affected. If it goes below 5, then fish will be irritated, or the acidic pH is not good for the health of zebrafish. Then third is the conductivity. Conductivity again is most important. So it can range between 100 to 1000 microsiemens, but conductivity between 300 to 400 is actually very good. We are maintaining our fish with the 300 to 400 conductivity for last 10 years. Okay, then you have to check the hardness of the water. When you get water from the bore well, then it's very hard water, so you have to soften this water using the RO system. But if it is a lake water or it's a corporation water, it is already soft water, so hardness should be between 75 to 150 milligram per liter of water. Then, as I told you, ammonia, nitrite, nitrate, and chlorine. In municipal water, chlorine is actually used for disinfection of the water. So this is again really toxic to fish. So you have to see, or you have to monitor these parameters regularly. Temperature, conductivity, and pH, you have to monitor daily. Ammonia, you can monitor once in a week or twice in a week. Hardness, once in a month. So these are the parameters you have to monitor while raising the fish.
Sir, last question, and which is interest of all. This is the question from Tanmay Patwardhan. He is asking how much approximately it will cost to establish small-scale zebrafish facility in institute.
Yeah, so I have actually suggested many pharmacy colleges in Mumbai, and even they have started a small setup using the glass tank. Okay, so I am telling you, for pharmacology studies, you can give a young farm student a simple, very simple experiments, like testing of any new molecule or herbal products. So in 96 hours, your studies are out. So for that purpose, you just maintain the zebrafish and just produce the embryos. In that case, even you can maintain the zebrafish in glass tank water, glass tank, or even rats and mice cages. Okay. So first you actually see whether these fish are actually survived in this setup. So you have to do some initial homework. It will take at least 3 to 4 months to standardize all these techniques, because everything you have to do, like water quality, you have to see all these parameters that I told you, like pH, conductivity, temperature, ammonia, everything. Okay, and once they are surviving in this setup, then you go for the breeding, whether they are breeding and whether they are producing good embryos, good qualitative embryos. If they are producing embryos, then you can actually use these embryos for your all toxicity or drug screening, simple experiments. If you have, so this setup will actually go in, it's not very expensive, maybe in 50,000, 1 lakh, you can set up in one simple room. But if you have funding resources, then one standalone unit can cost around 12 to 15 lakh, one single standalone system. And these are customized. So they can, like one standalone system, what we have, there are 12 10-liter tanks, and then the 36 3-liter tanks, and 20 1.5-liter tanks. So you can customize. You can actually ask them to make smaller version. So the cost will be reduced. Like if you need only 10-liter tanks, 10 or 20, or so that size will be reduced, and maybe cost will be reduced. Or if you want to make 3-liter tanks only, so these options are available. These manufacturers can actually do that. So, but if you go for the full scale, one standalone unit, it will cost between 12 to 12 lakh rupees. And then if you have no funding means problem, then you can go for the multi-leaking system, that what we have. So our system cost around 2.5 crore overall cost in 2012.
Okay, sir. Due to some time constraint, we couldn't able to cover all questions, sir. If you permit, we can.
They can actually write me. My email is there, so they can write me regarding all technical information for raising the fish, or quality control, or import and export. I can help them. Yeah, whoever is interested.
Okay, sir. So those who want to ask, okay, sir.
I hope the audience which are actually using the LibreOffice facilities or the LibreOffice model for their ongoing experiments, they will be benefited, or those who are actually planning in future may be benefited. So thank you once again for all.
Okay. So those who want to ask question to Dr. Kole sir can communicate with him via email. We will be sharing the email ID at the end of the session, also in the various group we have created. Okay, so just summarizing the sir's session, I'll say that in today's session, sir has imparted us the information about zebrafish as an youngest animal model in biomedical research, the origin, natural habitat, housing in laboratory environment, useful biological features, and that makes them suitable in many areas of manipulation. Thank you, sir. Thank you very much for giving your valuable inputs. We would like to see you again with us, sir.
Thank you. We'll see you during next IAC meeting.
Yes, sure, sir.
Yeah.
Thank you, sir.
Okay. Thank you all.
So with this, we have finished with our third session, third speaker. Moving on to the last segment of our session today, the vote of thanks. It's my honor to present the vote of thanks on the occasion of national level webinar organized by our college, MET BKC IOP. We have been fortunate to have the renowned identities from the industries. On behalf of my college and the entire management group, I offer hearty gratitude to all the speakers for sharing their graceful words with us in today's session. I am very much thankful to Honorable Sri Chhagan Bhujbal sir, Chairman Mumbai Education Trust, our respected trustees Sri Samir Bhujbal sir, Sri Pankaj Bhujbal sir, and our Chief Administrator Dr. Shefali Bhujbal ma'am. I express my sincere thanks to respected Dr. Sanjay Seshagar sir for being a motivational support to conduct such informative sessions. And this is all for the well-being of students as well as for the faculties. I extend my sincere thanks to HR department, EDP department, Marcom department, and maintenance department for every help they have extended us for the successful conduct of the event. Last but not least, I am very much thankful to our team members, MET IOP, all teaching and supporting staff for their every support and help associated with the program. With this concluding words, I declare that the session is over. Thank you. Stay safe.
Yes, ma'am. Thank you.